Bueno, bon dia a tothom. Anem a començar. Disculpeu, no us heu equivocat de jornada. Aquesta és l'anunci del míting que hi haurà després de l'estiu, al setembre, de cèl·lules dendrítiques. I, penseu que esteu tots convidats. Serà un míting molt interessant i és una miqueta, si voleu enviar abstracts i comunicacions, ara és el moment. Per començar, agrair-vos a tots l'assistència, els que esteu aquí i els que estan connectats online. Dic que estem molt contents perquè respecte del anterior meeting, el winter meeting, estem doblant l'assistència tant presencial com online. O sigui que confio que les expectatives que s'han creat sobre el meeting estiguin a l'alçada de les expectatives i que sigui un èxit. A part d'agrair-vos a vosaltres l'assistència, també agrair als nostres sponsors, que són una part molt important per poder fer aquestes activitats. Agrair a Standard Biotools, a MacBeck i Labclinics i a Pàlex. O sigui que moltes gràcies als nostres sponsors. Quan fem els cafès i això tenen els seus stands i podeu anar i veure una miqueta les coses que ofereix. I ja comencem sense més. Núria serà la presentadora de la primera comunicació.
So good morning, everyone. It's really a great pleasure to have the chance to introduce to our speaker today, Carolina de la Torre. She's the head of the core of the Proteomics facility that is located at the Institut Josep Carreras, that researchers on leukemia. And I think that she's been really one of the persons that has taught me the most in the past years in relation to proteomics and how its power can enlist the discovery of really unknown fields. So I really hope that you can enjoy her talk as much as I've been enjoying in the past year. So thank you so much, Carolina, for being here. And the floor is yours. Although I don't know where your presentation is. Do you know how to? No, le han dejado de compartir. Sorry about this.
There were a few. This was nice that they were going to buy, and they were saying that if there is an article from our end, we buy.
Okay. Good morning. Thank you for the organizer for inviting me today and give this talk. I will try to give an overview about proteomics and how proteomics can contribute in the last years to discover the malignancies in different kinds of diseases and either in health conditions too. And my expertise is not related to immunology, but I have been involved in different kind of projects where immunology is one of the main goals of the disease of the study. Our group is focused in the study of proteomes and proteomics, and we applied the last technology in order to answer the different kind of questions related with different kind of disease. As the advances that the proteomics has taken in the last year led us to know in more details about how the protein can be altered in some specific conditions. And so this protein can be altered in communicated with other. And for example, if everyone say, we can't see the sense that a proteomics can give the same information that gene expression said, give us. And gene expressions only express or can reveal the transcriptions phenomena that occur in the cells. But when we talk about proteomics, we only study the proteins and how these proteins are altered. Not only is this present or not present, or is upregulated or downregulated. We also can have the capacity to know more in detail about the interaction with each other that is really relevant in different kind of diseases, and as well the PTNs, the molecular post-translational modifications that are relevant for the protein functions. And in the last year, that can be impossible to do all the analysis of the proteomes at the same time. As well, when I started with my PhD, I working with 2D gels, and I make many 2D gels in order to see so difference, and the equipments don't have the sensitivity that now they have. And we can be able right now to identify with less than 1 micrograms of proteins, a lot proteins that can give very high and deep resolutions of the proteins in different kind of conditions. This is the tool that we have to answer and help to the researchers to design. We work together with the research in order to give them the best support that help them to answer the question that they have, because not always is, I want to do that, is exactly what you want to answer. As depend of that, you need to select a specific source of sample, because if you want to see the molecular alteration in some tissues that are have metastasis or aggressive, you need to see that probably the secreton or the liquid biopsy is the appropriated, but close to the tissues where is are damaged. Okay. Now, I would like to show a typical overview that the scheme that we use in our platform to analyze proteins in the high throughput way. First, we start with the sample preparations. Okay, this is typical. Okay, you do the protein extractions, but not all the samples have the same procedures. Depend. If you analyze it, a liquid biopsy, first you have to take account that the samples you normally can keep a 4 grade. No, because the platelets can be activated and secreted some cofactor that can be alterated during the protein profile expression. Either the saliva is a very fashioned liquid biopsy that now is useful a lot for discover precision medicines. But if you cannot use the correct way in the correct time, the enzymes can degrade all the protein that are expressed there. Different kind of tissues, cells, and do the protein extraction now, we use the specific regions because now we try to reduce time to be more close to the clinic, and we reduce the to aggressive regions that can let us to use a very manipulation procedures. And either for the digestion peptide, before we only do the digestion with the trypsins, that is the most common enzymes for cover the completely the sequence of the protein. Now we use the combinations of different kind of enzymes in order to give the best approach and cover completely the whole protein. Then we purify the peptides with the C18. This is another kind of advantage that we have right now, because we can do at the same time more than 96 proteins purify at the same time and inject directly to the mass spectrometry. And we use the last generation of the mass spectrometry. Before, we always have using data-dependent acquisition for analysis in the mass spectrometry. Now each time is more fashion to use data-independent acquisition, because you can be able to analyze the protein that are in the low expression profile. And that gives a big overview about the protein expression in general. Data analysis is the last but not the less important, because depending how you can do the analysis of the samples, you can take the wrong results or you can give the right results. You cannot analyze cells in the same way that you analyze clinical sample. Clinical sample, you have to take account different kind of variability, because each individual is completely different. And you take to take account different kind of variables, clinical variables that you have to include in the algorithms that the bioinformatics do the analysis, because that is personalized medicines. And we work in that direction. And you cannot analyze in vitro cells and the proteomes of the in vitro cell at the same way that you analyze liquid biopsy, because liquid biopsy reflects the protein that are secreted for some tissues, and the proteomes of the cells in vitro or the different kind of tissues reveal the completed proteins that are expressed in that cells. Okay, when you some researchers give me a stroke, I have to analyze. You expected that all the strokes have the same composition, but the stroke don't have the same composition, because some of them have more red blood cell, others have more white blood cells, or more different kind of protein. And you have to include all these variables, analyzed in the different ways in order to get a very nice results. Okay, all these ideas not comes only from us. They is the ideas that the National Cancer Institute inspired us for the clinical proteomic tumor analysis. And we are the only lab in Spain that be part of this consortium. The way is that we integrated the workflows that they use it in order to give a precise results. And we integrate not only the analysis of the proteomics, we integrate the clinical data, and we integrate the genomics data. This is the only way that we can really say that we are advanced in the precision medicine. Continue with that, as I said before, in the last 2 years, I am focused in the precision medicines, our lab. And we are working in a standardized the use of the non-invasive samples for the study of the proteomes in clinical studies. But when we started to do the analysis, we started with a plasma sample. But when we analyze plasma sample, we have to take many variables. Depend of what kind of plasma is. Is it for ETA? Is it for citrate? Or whatever. The result is not the same. We did many tests, and it's not the same that we reflected. Is it serum or is plasma too? And as well, if you use saliva, you have to take account that we have a lot of contamination with bacteria. And the only way that you have is to recollect quickly in the short times, put inhibitors, or keep at -80 until the analysis. As well, we can analyze the whole saliva. But first, we need to eliminate the bacteria. And as well, it's better, but the different kind of study that we have done is to analyze the exosome of the saliva, because that is a really relevant sample source for analysis. Okay, now I will show the different kinds of sample that we make the studies in collaboration with some researchers. That is one of our last collaborations with the doctora Nuria Izquierdo from IrsiCaixa, that was for know about the mechanism of some drugs as antiviral functions with SARS, and we want to know more in detail about how this drug acts in the cells and how this drug, the mechanism of action that they have, not with only with the proteins related with the virus, but also with the proteomes in the host cells. And that results can be able to show a deep analysis of the molecular pathways where the drugs are taken inside to the cells. And this, I think it's almost to submit that paper. It's a really nice paper, and it's just a little bit summary because I am no expert in virology. But the nice things, I take this little example because it was impressive that in different kind of dose of the drugs, we could see how the protein expression from the virus can be reduced, and the other spike protein, yes, this. Yeah, no. And how the protein, and here from the protein, from the upregulated protein from the virus is upregulated when there's no concentration of the drug, and when is the drug concentration of the cell treated with the drug is reducing completely the upregulation of the viral proteins. Sorry. This is other examples, that's other projects that I did many years ago, was during my PhD in São Paulo Hospital. And I like a lot this study because it was at the first, more than 10 years ago, when the high throughput technology was not used, but the two-dimensional gel and Western blots reveal a very nice technology to identify autoimmune peptides that are not established in that moment. And we can be able with this technology to identify a new antibody that some patients with polyradiculopathy have against the mutant protein 2. And it was a really nice study where 2D gels from the extracts from the nerve road, anterior and posterior, we can perform the analysis, and then we did the Western blots with the serum of the patients, and we can identify this molecule. This molecule is isolated, was digested, and we reveal which protein was the interest. To validate these results, we synthesize some peptide and we perform the ELISA to see the concentrations in other patients, and we validate that exactly is this protein that was associated to that. Okay. Continue with the sample of an application of the proteomics along the years have helped us in different kind of diseases. Now we continue with neurology, and this is more in the study in collaboration with the Hospital de Belviche, and is to discover the molecular composition of the stroke in the etiology of the cardioembolic, atherothrombotic, and cryptogenic. Cryptogenic means that the clinician cannot be able to identify the source of the etiology of the disease. They are not 100% sure is this cardioembolic or thrombotic, and they have doubt. And depending on how doubt they have, they cannot give the right treatment to the patient. And we did the protein extraction of each stroke. We analyzed that, and as they say, we have 30 patients from the cardioembolic etiology, 30 for the atherothrombotic etiology, and 20 from the cryptogenic. And when bioinformatics analyze the results, doesn't reveal any substantial results. What happens is that it's a shit. We need to do again the analysis. And then we say, okay, that patient have a lot of comorbidity. We need to do an unsupervised because talking with the clinicians, they say, no, this is positive cardiobolic because they have atrial fibrillations, because the patient have a heart attack 1 month ago. But it's not precise the way how they say that the etiology is cardiobolic or atherothrombotic. And we did that, and in supervised ways, we analyze the samples, and that reveal amazing heat map where we can be able to detect 4 groups. And what we did was talk with the clinician again and say, hey, we have identified that, what happens that, and we try to integrate all the clinical information and the clinical tests to give the complete overview about that if these results are trust or not. And we take another data that are in the different kind of database from the stroke, and we did the analysis in the supervised way, and we confirmed that these subtype are present. Now what we did, we are doing right now is to do the validation. First, we did the validation in the plasma, was the some key proteins that are involved in these 4 groups, and we did the analysis in the sense cohort that we did this analysis, and now we are doing in the big sample cohort. And we expect the soon to take a very nice result of that. That is the summary of this study where we identify these 4 groups. And in the first groups is more related with acute phase response. The patients are younger than the other ones, but have apolipoprotein super alterated and many protein related with stress or infections and other things that cause the thrombus. As you know, when COVID pandemic, many persons, many individuals have problem with a stroke, and is this control of the response of the immune system that can cause some alterations of the way. And the other group have more related with the chronic inflammations and toxifications and catabolic process pathway. The other is more related with the functional mitochondrial, and the other one has more in common with the group 3, but as well have more protein related with death and necrosis. Okay, this is the conclusion that I said. That is my group and the Department of the Neurovascular Disease in Belviche. Now we move to the cancer. This study was done with keratinocyte cancer in collaboration with the Institut, the research in Lleida. And we have identified here some specific biomarker from the keratinocyte carcinomes. But the most relevant here is, as I said in the previous slide, is what kind of sample source we use for that. The researchers was the main objective of they was to identify the protein profile of the associated to the malignancy of the disease. The most common keratinocyte carcinoma are the basal cell carcinoma and squamous cell carcinoma. But squamous cell carcinoma have higher malignancy and have a lot of metastasis. And in order to identify the difference between both, I think that the best way to analyze that was with TIFF, that is the tissues interstitial fluid. What samples they have? They have a biopsy from the skin patients. And if you analyze the wall tissues, you cannot be able to decipher well which is the protein that are secreted or not. And what we did was to extract the tissues, interstitial fluid, and we optimized the centrifugation condition, blah, 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 in order to get exactly the source of biomarker that can give us the real information about the malignancy of that disease. And this is an amazing piece of data where we see here the different kind of samples that we have analyzed, and all of them are completely well separated. And the heat map also shows the clear difference of the teeth from the BCC and SCC, either the molecular way. And then what we did was with the pathology department, we did the validation of the candidate and was well matched with the previous results that the proteomic have discovered. Is in that case, that's as the conclusions, what say that the proteomics reveals exactly the proteins that are associated to the malignancy of the disease. In the case of the squamous cell carcinoma, we identified that upregulation of the protein related with the cytoskeleton, like stratafins and latinins, and that in other cancer was related with the progression of the disease. And the teeth of the squamous cell carcinoma also was enriched with these cytokines that are well known, that are associated to the aggressiveness of malignancy cancer. That study was in collaboration with the SCI lab from the JEDA and Department of Neurology and Department of the Pathology. And this is the last one. This is, we have 2 years doing the collaboration with the Barça. We analyzed the sample of the female elite football player, and we analyzed from there. Has to be a non-invasive sample because they need to don't distort so much, and is try to, in the less time, give the best result of the analysis. And with this sample, we have been able to optimize all the procedure for the epsosome analysis from the saliva. And the first aims of this study was to identify some proteins that can predict the risk of injury or the fatigue, or how we can improve the performance of the players. And surprisingly, we identify 30 individuals, elite women. And what we did was based on the CPTAC consortium. What we did is that how you can to match this group of individual recipients, no? Because you don't know if these have a very good performance, have injury. We take account all the information that the clinician and the physician says us. But of course, each of them take supplements and many things that either we have to integrate in the result that we get, no? And what we did was the unsupervised analysis and see how they can be organized. We see different kind of pattern of the individuals sport that we have analyzed, and they showed clear difference. Some of them is completely different to the other ones, and we have to make a zoom of C alone in order to see more difference between the other ones. And the other way to do the analysis was to say, okay, we have all this group. Now I want to see what is the more relevant pathways and functions that are represented in the data set that I have. And we identified these 5 clusters of protein. That was the analysis for the Pearson correlation that related to how is not only how upregulated or downregulated. Is how all these proteins that be part of some pathway are represented in this liquid biopsy, because it's liquid biopsy, again, is secreted. And we want to integrate both informations, and with the integration of those informations, what we get is here is the 5 cluster of proteins pathway that we have identified. And here is the group of the individuals, females that we can, that the proteomics study revealed. When we integrate both, say, the first one I say is not real nice, but in theory, all of them are healthy. And we have to evaluate each one how is in this cluster of protein is related with lipid metabolism, energy production, blah, 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 no? Okay. These groups are classified and we see upregulation, but no this cluster of protein in the same level. But we see that, for example, in these two, we have upregulated. Okay. Continue with the second one. That this is the first individual that she have a special pattern different to the other one, and she have a lot upregulated protein related with mitochondria. And okay, talking with the physician and the doctor of the team, they say, okay, that is more athletic. Is the metabolisms that individual are more related with anabolic. We started to talk more in details about why they have upregulated these proteins and so on. And here we see also that group has upregulation and these other groups. The really nice thing was that when we came here, that our protein are related to the actin filaments organization, that is that these upregulated protein that are there in this cluster are associated to the individual that have with fatigue, that have injury in some muscles or whatever, that are recovery, and was, okay, that is really good. We need to go in detail about exactly which protein are expressed there, and so on. Now, well, this is the summary of the results, the previous result. But now we are analyzing the last study with TENS is in order to optimize is when we have the genomic, because that study is in collaboration with them, but we have to be able to integrate the genomics data of each one. But of course, is to do more than 3 timing collection during the season. And now this year we have collected during 2 days a record of after training, before training, after, and I think that 24 hours after too, yes. And it's to see the protein profile, how changed when are in relaxed way after the trainings and after 24 hours. And this is what we are doing in working with personalized medicine and proteomics. Thanks.
Any questions?
Okay. So I'm just curious to know how you work. So a researcher will contact you and then you prepare a project, or you work like a facility where somebody can send you samples? How is it?
Depends. We do everything. Now is depend. In some cases, we help to the researchers since the beginning to write the project, and we are part of the project. And the other way is like that, is you only want a service, a single service. Okay, we do the service, and that is. But when we're working in some projects like personalized medicines, we have to invest many times in the data analysis and the optimization of the sample, because now you see, nice the results, but we are not a big group. We are a normal group that we grow up with many work, and we work in the optimization of the sample collection and everything. And not all the things that we see in the paper always is nice, because now in the reality, when we are doing the analysis with the clinical samples, is like something that caught me the attention and surprises is we don't identify the same proteins. I take your samples with EDTA and citrato, and I don't have the same protein profile. And either I analyze the samples of the pack of do the cells, and probably I detect more in citrato of you, and then I detect more in ETA. But it depends on the profile that I have. But I discovered that after doing tests from control collection. And what we really want is not to do only a one single service. Is to work together and help the research since the beginning, because sometimes you write the paper, you write the project, but probably you are not using exactly the right experimental design. And you can get in the easy way, because the proteomics is taking advance every year. And now it seems impossible, but we can get results in less than 2 or 3 days. Before that's it was impossible. I don't know. Nuria can explain more in detail, because I collaborate with her since the last 3 years and in many projects. And
No, I think that you give a very added value just by the fact of helping researchers to understand which is the best pathway to perform your proteomic analysis. Because this is something that probably you might not know when you start the project, but they have always been very helpful in guiding us through the way. And I think it's very important, because you might find problems. And if you have a problem, you need an expert to solve it. But I also wanted to ask you in relation to the possibility of doing single cell proteomics. I mean, this is something that is becoming...
we have started with that. But when we don't have the machines, and was a little bit challenged. Now we have some toys that we can try to do. But depends what you really want to see, because only one is single cell, or you want to see an isolated group of a few cells, because I am more realistic. Now we are more close to that, and I can give abstract results of that one single cell. Probably with the astral we can do, but I don't have the astral at the moment. Matthias might have, and I think that other one. But I did a test with some research in our institutions, but it was with 100 cells. I think that we started to do when you was there, that some guy from the Alex Vaquero goes to you to do the isolation, and I called you to say, hey, I cannot add to BSA, because the BSA can contaminate the cells, and I will see only BSA, but they need to do the sorter, you need to put a little bit BSA, no, I remember.
Single cell, and then go to Caterina. For the
Yeah.
for not for
Yeah. But the key is work together. And one thing that I think that gives more value to our platform is that we don't give you the list of protein, because when we give the list of protein to the clinician, they become crazy. Oh, okay, what is that? And we try to help, and we try to tailor the results and discuss with them, okay, we have that. This is related with that pathway. And I see that this is the value, because each one is expert in something. And I need the information of them, but they need the information of us. Because sometimes when I'm talking with the neurovascular department, I am like here and say, okay, but what is that cryptogenic or whatever? And it's the same way when I'm talking about protein, and probably we have a very amazing result, but we cannot be able to understand well because we don't talk the same language. And the good thing is to integrate everything.
Any other questions? Thank you very much.
Thank you.
Sí.
Okay. Good morning, everyone. Our next speaker is going to be Victor Jiménez. Victor studied biochemistry in the University of Barcelona. Then he did his master in the University of Vic, a master related with omics data analysis. And currently he's doing his PhD under the supervision of Dr. Iñaki Martín Subero in IDIBAPS. So whenever you want.
How do I hide this? Okay, so good morning, everyone. First of all, thank you for inviting me today. My name is Victor, and I'm a PhD student in the Biomedical Epigenomics group at IDIBAPS. And today we'll talk to you about single-cell technologies and their applications in the study of immune cell populations. So let's start with the very beginning. It doesn't work. Okay. To introduce you to the concept of single cell, I will use one of the most widespread analogies, which is imagine your sample, any given tissue, tumor, organ, anything, is a fruit salad. In this fruit salad, there are different pieces of fruit which represent the different cell types, and each one will contribute to the salad, to the properties of the salad in a different way. You want to study your salad at a very deep level, that is, you want to study your sample at the molecular level. So one thing you can do is a milkshake. The milkshake will give you a general idea of the flavor and will give you an idea of how the fruits behave as a whole. This represents bulk sequencing or massive sequencing. And with bulk sequencing, what I'm saying is that you obtain the average levels of gene expression, accessible peaks, chromatin peaks, any given molecular layer. And this gives us a robust view of the molecular landscape of your sample. With this, we can do comparisons, we can characterize the sample, we can discover new biomarkers, but it is not the best if what you want to do is know more about the fruits that make up your salad. So that's when single-cell sequencing comes into play. And with single-cell sequencing, you obtain sequencing information for every separate cell. This allows us to study deeper the cell composition of your sample, the cell dynamics, the heterogeneity, and even discover new rare subpopulations. You might discover new flavors that with the milkshake get a little bit masked. So maybe here you are thinking that instead of single-cell sequencing, you could apply techniques such as flow cytometry, and then with the subset of the isolated cells, apply bulk sequencing. But here, however, single cell is agnostic in the sense that you don't need any marker, any known marker to isolate any given population first. And besides, maybe there are some rare subpopulations that are so infrequent that applying bulk sequencing is a problem for them because they are so infrequent. And finally, we can also talk about spatial omics, which would be kind of combining a near single-cell sequencing resolution with the spatial information. With this, you can study the compartmentalization of your sample, where the cells are, the cell-to-cell interactions, and even the trajectory, the developmental trajectory of the different cell types. So in the last years, single-cell technologies have increasingly evolved to cover 4 different omic layers. And to mention them, I will start with single-cell DNA sequencing, with single-cell copy number variation, single-cell whole genome sequencing, and more, that allow us to study the somatic mutations in each cell and thus uncover the genetic heterogeneity. For example, the somatic mutations across clonotypes in a given lymphoma. That is what we do in our lab. Also, of course, a single-cell RNA sequencing with different modalities that allows us to characterize the different cell populations and their dynamics, and uncover the gene expression patterns of the different cells. Epigenetic insights are also available with single-cell ATAC-seq and that allows us to gain gene regulatory network insights underlying the gene expression patterns that we obtain with the single-cell RNA-seq. We also have single-cell epitope indexing with technologies such as CYTOMICS that give us information about protein abundance, but more specifically cell surface proteins. And lastly, I want to mention also single-cell BCR and TCR sequencing, which allows us to do a very deep immune profiling of every single cell and study the defined clonotypes and their evolution. Of course, there are a lot of other single-cell technologies that I have not mentioned. Specifically, I want to mention the epigenetic technologies that are out there, like single-cell methylation and single-cell chromatin profiling techniques with single-cell ChIP-seq or single-cell cut and run. And although they are not still so commercially available, I think we will hear more and more about them in the next coming years. And also, I would like to mention that there are combinations of these kind of technologies to obtain a multi-omic profile of the same single cells at the same time. So what can we do with single-cell data? Being illustrative, if you look for the term cell atlas in PubMed, you might get an idea of one of the most widespread applications. And in fact, single-cell atlases have filled the field with enormous efforts and huge international consortiums, the main one being the Human Cell Atlas. This project arises with the goal of understanding and mapping every cell type in the human body using single-cell technologies and spatial omic technologies. The Human Cell Atlas encompasses more than 1,700 institutions over 90 countries and has helped mapping this our human body, the cell types in our human body, with more than 400 projects and divided in 18 different biological networks representing the different parts of our body and processes. And it is important because these resources are available for the community. So this data, you can access this data through the Human Cell Atlas portal. And here I included two examples of immunological atlases, two different immunological atlases that help us uncover the immune heterogeneity within our body into different tissues. This one, the Human Cell Atlas, I will explain you later. So single-cell techniques go beyond this rather descriptive research with annotating and defining cell types. And to illustrate that, I brought to you two different examples. The first one being this study made by our program at IDIBAPS that focuses on Richter transformation. Richter transformation or Richter syndrome is an aggressive progression of chronic lymphocytic leukemia into a diffuse large B-cell lymphoma. And in this study, single cell helped confirming the presence of Richter clones here in these figures in samples way before the diagnosis of Richter, and also helped constructing a gene expression signature that differentiated the Richter clones from the normal CLL clones, from the normal chronic lymphocytic leukemia clones. And this gene signature was in accordance with other experimental findings showing a higher OXPHOS activity and a lower BCR signaling. But about these things, Dr. Heribert Playa will tell you more about in the following NAIVE immunology session. So the second example that I wanted to bring you is this preprint study in which they use single cell to characterize different transcription factor networks related to different B-cell maturation states, and then quantify these transcription factor networks in different mature B-cell lymphoma samples. So here you have, for example, the abundance of the different B-cell subtypes in different types of lymphoma. Recapitulating a little bit, single-cell technologies are a good option if what you want to do is study, know more about the cell composition, cell dynamics, and heterogeneity of your sample at a level of granularity that has no precedence, both in this descriptive manner and to gain insights into the biology in health and disease. Also to mention, as I have been telling you, that there are a lot of technologies available, and it is a bursting field where new computational tools come out almost weekly. So there's a lot of movement and activity. But I have to say that it has some drawbacks too. And the truth is that the data are yet rather expensive to obtain, and they are quite heavy, space and time consuming, computationally speaking. Since it is a relatively new field, there are also a lack of standard robust procedures in some parts of the analysis, but the most limiting issue in single cell is the sparsity of the data. That is, you will be lucky if in single cell RNA-seq you have a mean of 3,000 genes per cell. That is, that if you have a matrix of cells and genes, most of the counts in every cell for most of the genes will be 0. And that is mathematically challenging to do the analysis. But the message that I want you to take home is that single cell is not a substitutive or a better version of bulk sequencing. It is another thing with other goals and other applications and other limitations. So now that we have the basics of single cell sequencing, I would like to dive a little bit deeper into the single cell analysis. And for that, I will start by presenting the 10x, the multi-omic modality from 10x Genomics. 10x applies a droplet-based technology in which the individual cells are captured by droplets that contain an oligonucleotide barcode that helps defining the cells later in the analysis. Once the cells are captured, the RNA and ATAC libraries are prepared and amplified, and thus we obtain transcriptomic and epigenomic, the epigenomic layers of the same single cells at the same time. I included here a picture of the real thing, the moment in which the drops capture the cells, and here a picture below of the 10x Chromium, which is the machine that does the whole job, and it is only about 30 centimeters, so it is nothing. So after the sequencing, there is an initial processing of the data, the FASTQs, etc., and then you obtain the count matrices or the peak matrices or whatever. So single cell requires an exhaustive computational analysis to get to the biological insights, which is what we are interested in. The first things that you have to do when doing the downstream analysis are the quality control and initial visualizations. For that, we use RStudio in our case with deeply established pipelines used in the community, which are Seurat and Signac, developed by the Satija lab. But there are also other environments available in Python and that are very used also. So the quality control has 2 layers. So the first one is the technical parameters, in which we filter cells according to parameters such as library size, number of genes, number of features, and in ATAC, for example, the nucleosome signal. But I want to mention importantly the percentage of mitochondrial and ribosomal genes, because in single cell, the signal of these genes is associated with cell death, and thus the quality of the cells to then do the analysis. For example, in this figure, I put here, you can see here a group of cells that display a high signature of mitochondrial genes, and not only the high signature, but these cells that display the highest signature clustered together. So we with this and with other parameters, we can remove them from further analysis. The other layer of the quality control is the doublets, which are artificial constructs that are done in this part of the procedure, in which 2 or more cells are captured by the same drop. And then we have cells that have a transcriptome that is not, that should not be there. There are mathematical algorithms to remove them, but you can also manually remove them. For example, if you catch a cell that expresses CD20, a typical B cell marker, and CD3, which is a typical T cell marker. This is not, well, we don't know about the existence of these cells, so we remove them in the mature stages. Then we start with the visualization of single cell data is not a trivial thing. And since it's such a high dimensional data where we have thousands of cells with thousands of genes, we have to apply mathematical algorithms to summarize this information in a 2-dimensional space where each dot here is a cell. For this, we apply the most used methods are UMAP and TSNIS, but these are a little bit tricky to deal with because they are not linear. They are not like a PCA in which you can say that, for example, this cluster would be more different than this cluster or this cluster just because they are closer or farther away, or you cannot infer directly a trajectory of these cells that differentiate into these cells and then finish here because it is not linear. To do this, you have to first clusterize the cells, analyze the clusters, and then infer the biological insights of the human. And I'm telling you this because every week there is a lot of controversy in the field with papers interpreting the UMAPs badly or inferring information just based on the UMAPs, and this is, well, not done. So now that we have the very basics of the single cell analysis, I will talk to you about some of the results that we obtained with single cell data in the tonsil atlas, which was published in February in Immunity. So as you may know, tonsils are secondary lymphoid organs that stand as the first immunological defense against inhaled or ingested pathogens. So having an exhaustive map of the cell subtypes and the cell dynamics and how they regulate themselves in different situations was an important resource for the community. So in this atlas, there were 121 cell types that were annotated and characterized with more than 550,000 cells sequenced from 17 patients. The design of the analysis included a discovery cohort, which was used for the initial mapping, annotation, characterization of the cells, and a validation cohort, which had curating and validating the initial results. This atlas was also a multi-omic atlas in which we had single-cell RNA-seq and single-cell attack-seq with the multi-omic modality, but also we had CYTOMICS, the single-cell epitope sequencing, and single-cell PCR and TCR sequencing to exploring the clonality, and also spatial omics data. So the atlas is really exhaustive. It has insights in different cell populations. I encourage you to read it, but for now I will focus on some of the results that we have found regarding the B-cell maturation states. So as a quick introduction, and as you may know, naive B cells upon antigen activation in secondary lymphoid organs with this T-cell interaction undergo what we call the germinal center reaction. In this germinal center reaction, there is a cycle of proliferation, somatic hypermutation, and class switch recombination in the dark zone and immune selection in the light zone that allow these naive B cells to differentiate into memory B cells and plasma cells. So with that, in the atlas, naive B cells, lots of different naive B cell subpopulations were identified. There were resting naive B cells, but also there was a gradient of early activation of naive B cells with the expression of CD69, that is, I can see it now, but it is here, with the expression of CD69, but also other mechanisms of naive B cells activation, such as interferon genes. Also, there were some signs through MYC expression followed by CCND2 expression of early commitment of these naive B cells to the germinal center. So it was the whole path of naive B cells to the germinal center. As for the germinal center, there were transcriptomic features that were identified beyond the proliferation, which is the main thing that happens in the dark zone, and they were related mainly to the re-entry of these cells from the light zone to the dark zone in a cycle that repeats itself. And finally, as for the latest stage of maturation, there were more than 20 plasma cell subpopulations identified, also following this trajectory from the precursors and early plasma cells to the fully differentiated plasma cells secreting different immunoglobulins, and also the trajectory from memory B cells to memory B cell derived plasma cells. Not only did we characterize different subpopulations, but also wanted to have a look into the regulation networks underlying the different subpopulations, and I will tell you about some of the key findings. So the first one is related to BCL6. BCL6 is a transcription factor that is related to the germinal center reaction regulation in B cells. But in this atlas, we saw through single cell attack data that the BCL6 also does this function through follicular helper T cells. And we can see this because here, if you look at this UMAP representing the T cells, you can see that the accessibility of the BCL6 gene is the same for all the T cells. But if we look at the accessibility of the BCL6 distal enhancer, which is deeply characterized in the bibliography, we can see that the follicular helper T cells have a higher accessibility. But since accessibility is not activity, we validated this with ChIP-seq data, bulk data, where we saw that the signal of H3K27 acetylation was higher in the follicular T cells. So BCL6 also plays a role in the T cells in the germinal center. Another thing related to the germinal center is the characterization of 3 different accessibility modules regarding the re-entry of B cells from the light zone to the dark zone to proliferate and repeat this class-switch recombination process and somatic hypermutation. So the first module was related to NF-kappa B genes that progressed to the activation of activator 1 proteins, and then finally a module related to the activation and regulation of 8 genes, which, as you may know, are related to the class-switch recombination process in the germinal center. Related also to the B-cell epigenetic reconfiguration, there were 3 different modules identified with different transcription factor families. The first one related to plasma cells with an enrichment in IRF motifs. The second one related to germinal center B cells with an enrichment of EBF1 and NF-kappa B genes. And the third one related to a more general B cell module, which was enriched in EBF1 and PAX5 transcription factors. And lastly, 6.5 transcription factor was identified as a novel potential regulator of plasma cell differentiation, as shown in the transcription factor activity, which is computed thanks to the combination of single-cell RNA-seq and single-cell ATAC-seq data, but not only the transcription factor activity, but also the upregulation of the target genes. This is something that was further confirmed, again, with bulk data, in which we see a higher gene RNA levels of 6.5 in plasma cell subpopulations, but not only RNA, but also protein levels, with not only in plasma cells, but also in multiple myeloma, which is a cancer derived from plasma cells. To end with the tonsil atlas, in the study, it was very important for us to make it a useful resource for the community, following the first principles in bioinformatics. So this came in the form of a glossary that explained the different cell types annotated in the study. Also, different ways to access the process data in a user-friendly manner so that other researchers can also have a look at the gene expression patterns in the subpopulations. And, well, also in the form of an R package for people that also do data analysis. And finally, ways to map or annotate other single-cell cohorts with the tonsil annotation. And it is very important because in our group, we are still working on different single-cell projects with chronic lymphocytic leukemia and mantle cell lymphoma, and the tonsil is standing as a control and a reference to map this normal B-cell maturation variability to the tumoral cells. And that's it for the talk. I would like to give a very big shout out to my group and the people who carried out the bulk of the project. It is a great honor to be part of this science. And also, I encourage you to look at the SciComm part of the project with this Welcome to Tonsil comic that summarizes all the findings of the paper. And thank you for listening, and I will be glad to answer any questions.
So questions for Victor? Yes.
So very nice talk, yeah, and very impressive results. So I'm wondering, so you identified a lot of subsets of plasma cells, and you mentioned the trajectory studies. How you deal with that? So do you have special programs to deal with the trajectories, or did you do that by looking at the results and trying to figure out from which subset you go to the next one?
It is a mix of both. There are some algorithms that do this trajectory analysis in which they mathematically compute how the genes get upregulated and downregulated through what they define as pseudo-time, because we don't have the time in these cells, but also through manual curation. That is, you have a cluster that expresses an early gene activated in early stages, and then you have another cluster that has this gene a little bit downregulated and activates the following one. So that allows us to define the trajectory.
And do you find a single trajectory, or there are different branches, or you can end up with at different ends, or how is
So-
it done?
As for the mathematical part, the tools that do this trajectory analysis are a little bit tricky because you define previously what trajectory you want. So it is kind of supervised in a way. But as for the manual correlation is what we know. So plasma cells can be memory B-cell derived, so or the independent path. So it is, well, you can obtain this branched path if you want, and if you know that it exists.
And then I have a technical curiosity because of the tonsils. So were the samples fresh or they were frozen? And if they were frozen, do you have then problems
They were
to obtain like a lot of that? If they are frozen, then you may have dead cells. And in our experience with the tonsils, if you freeze down the tonsils, sometimes you lose some of the germinal center cells, the bigger lymphoblasts. So how you manage that? Yeah.
They were fresh, and but there's a previous study that also did this like technical exploration of the differences that might arise in single cell data with frozen samples and not frozen samples, and the genes that might get activated like in a technical way. But they were fresh. And as for the germinal centers, it is true because most of the samples here were from old adults, and also we had the problem with that in old adults the germinal centers are smaller, so we had fewer cells of the germinal center.
Questions?
More according with the question that you did, is that you did the validation of the viability of the cell when you do the analysis previously?
Yes, it is experimentally assessed.
Because
Before the-
continuing with that is we did the analysis with some of the collaborator, and they do single cell genomic transcriptomics, so on, and they give a little bit part of the isolated cells that was from the lymphoma samples, and when we analyze it, we see different, clear difference between the group, and no were associated to other things, that was only to the viability of the cells. But genomes analysis, you could not detect that. But the proteomics is that proteins that are related with, proteins that are related with threats or malignancy.
If you have single cell RNA sequencing, this is relatively easy. So we assess the cell viability experimentally before the sequencing, but then of course if you do flow cytometry before, also you can isolate the cells that you are interested in. But of course, if you are dealing maybe with, I don't know, experimental model that makes the B cells proliferate a lot, you will have a lot of that. So you use these technical parameters like the mitochondrial genes to remove these cells, and so they will not be part of the comparison and will not influence the results.
Thanks.
Any other questions? I have a very general question for you, Victor. Thank you for the nice talk. You mentioned before that the Human Cell Atlas is a very good resource in order to find many data sets for many tissue samples from human. So my question is, if a researcher is interested in integrating different data sets from different studies, what do you think is the best way to remove this batch effect that may appear in the analysis?
So one thing I would probably try to check is that the cells come from the same technology, in the sense that if you have an atlas of 3' single cell RNA-seq, the other atlas that you want to combine is also 3' single cell RNA-seq. Because normally the technical procedure that you follow is one of the most variabilities. But I did not show anything, but like the most strong variability is the patient or the in healthy tissues not so much, but in tumors you see each patient is a cluster, separated cluster. So for this in single cell RNA, in single cell data, there are different programs, there are different algorithms that allow you to integrate the data and remove this variability bar for sample. But normally when we integrate different projects, we do it by sample, because that way you also remove the technical variability of the procedure followed.
Okay, and also you also talk about the sparsity of the single cell data, and it's very common that you can find in the cells some genes that are 0 expressed, let's say. So do you think this is a biological thing or more a technical issue?
So we started with that a lot because So with single-cell RNA-seq, it's so sparse that you are in the frontier between detection, no detection of the genes. So the genes that have counts, they have very few counts, and you have to work with that. So most of the zeros, you cannot tell if a zero is because there's no expression of the gene or because it was not captured. Sometimes you can infer if it is not captured because maybe a specific cell has a zero, but another cell have a one. So if you look at the cluster, you will see some certain level. You rely on the group of cells, not on the individual cell. So yeah, you have to, it is an unsolved question in this way.
Thank you. Any other question? Thank you.
Ahora tenemos un... Tendrá que ser un poco más corto de lo previsto, pero bueno.
In English, so you can also add a question in English as well at the final of the presentation. So, vamos a hablar de citometría espectral. Lo primero de todo es aclarar conceptos. La citometría espectral al final da la sensación de que estemos hablando de algo como muy novedoso, aunque hace años ya que se inició. Suena como a película de Star Wars. Pero realmente, y el concepto de citometría espectral, que es lo que os voy a explicar, no es del todo correcto. Tenemos que hablar de citometría de espectro completo, de full spectrum cytometry. ¿Por qué? Porque la citometría de flujo siempre ha sido espectral. Terminamos midiendo dispersión de la luz, fotones a partir de compuestos fluorescentes, o sea, la citometría siempre ha sido espectral. Lo que pasa es que de la citometría convencional a la citometría de espectro completo hemos tenido un cambio. Claro, en la convencional, nuestro principal interés estaba focalizado en detectar una parte de esa señal de luz. Y es como si viéramos un paisaje a través de una ventana. Lo que realmente se termina viendo en la citometría de espectro completo, y os puedo asegurar que esta foto no está sacada de Google, sino que Hamad la sacó en su visita a Chile. Hamad es mi compañero en el clínico en la parte de citometría espectral. Y al final lo que nosotros terminamos observando en la citometría de espectro completo es mucho más, aprovechamos mucho más las señales fluorescentes de lo que se hace en citometría convencional. ¿Por qué? Pues para que veáis un poco, si un fluorocromo muy tradicional como la ficoritrina, pues nosotros disponemos de filtros de paso de banda para seleccionar la longitud de onda proveniente de un láser concreto, pero esto no es realmente la foto completa. Al final, lo que terminamos detectando en los fluorocromos y lo terminamos viendo en un detector primario, el principal, pero tenemos señales secundarias que en citometría convencional no utilizamos, o más bien nos estorban, y las eliminamos con la llamada, con la spectral compensation, o sea, con la compensación de señales fluorescentes, y esto en convencional no se usa. Por lo tanto, una de las primeras diferencias es que en citometría convencional, si tenemos un detector de señal fluorescente, ese va a ir asociado a un fluorocromo concreto que tengamos en nuestras muestras en nuestro panel. Pero cuando hablamos de citometría de espectro completo, fijaros que los pasos de banda de los detectores van a ser muchísimos más en los equipos de espectro completo. ¿Por qué? Porque la idea es recoger toda la señal de luz que se producen o que es derivado de nuestras células y por el paso de cada láser. Con lo cual, al final, no tenemos huecos entre esos filtros de paso de banda, exceptuando las longitudes de onda de los láseres. Por lo tanto, si nosotros en nuestro laboratorio tenemos varios equipos, como podéis ver este ejemplo, pues vamos a fijarnos sobre todo en las dos últimas filas, en los fotomultiplicadores que recogen la luz de fluorocromos, porque se utiliza uno o dos para el cutter. Si en el Fortesa, por ejemplo, tenemos 18 detectores de fluorescencia, el número máximo de fluorocromos que podemos leer son 18. Parece malaria, pero si tenemos un equipo espectral, el número de detectores, en este caso o en este ejemplo, son 64, pero eso no significa que podamos utilizar 64 fluorocromos a la vez. Podríamos llegar a utilizar 90 o 100 si cumplen lo que os voy a contar a continuación. No hay una relación numérica en cuanto a los detectores. Este sería un ejemplo de un equipo de citometría de espectro completo. No voy a entrar en detalles. Y este espectro completo se recoge toda la información para generar lo que se llama la spectral signature o la huella espectral. Y al final vamos a tener un rango de longitudes de onda. Vamos a ver, dependiendo del fabricante del equipo, lo vamos a tener con más o menos colorines, pero esto al final es una representación de lo que sería una media o mediana de fluorescencia de cada uno de los detectores. Con esto se termina fabricando una huella espectral. Y esto es el espectro completo. Entonces, a partir de ahí, tenemos que hablar de fluorocromos. Es decir, los fluorocromos tienen una huella espectral cada uno característica. Es decir, ¿cómo se parecen entre ellos? Entonces, aparecen, casi todos los fabricantes suelen trabajar de una manera muy similar, de tener valores entre 0 y 1, que el 0 es que cuando dos fluorocromos tienen un índice de similaridad que es 0, es que son absolutamente diferentes. Y es 1, es que son prácticamente similares. Y con esto conseguimos ver las propiedades de esos fluorocromos para lo que va a venir después. Obviamente, cuanto más láseres y detectores, sobre todo láseres, tenga tu equipo, vas a ser capaz de diferenciar mucho mejor estos fluorocromos. Un ejemplo. Dos fluorocromos que son incompatibles en citometría convencional. El Brilliant Violet 421 y el Super Bright 436. Tienen un índice de 0,94. Quiere decir que sus dos huellas espectrales coinciden en un 94% de esas huellas. Y lo que tenéis aquí abajo es la huella espectral. La huella espectral del BB421 y del Super Bright 436. Vais a ver que tienen dos zonas que son muy similares, pero no del todo, que es la zona que está rodeada en rojo y la zona que está rodeada con el círculo azul. Bueno, ¿esto qué nos permite? Porque lo de las huellas está muy bien, pero los que hacéis citometría lo que queréis ver son los plots. Y entonces aquí tenemos dos ejemplos de dos fluorocromos que son incompatibles en citometría convencional. El BB421 de Super Bright, que es el que os he comentado antes, que es el plot de la derecha. Y fijaros que tenemos, en este caso, un CD123, que marca principalmente poblaciones de dendríticas, basófilos y CCR7. Y son absolutamente compatibles. Hay una buena resolución para diferenciar los patrones de expresión en unas células, en este caso, son células de sangre periférica. Pasa lo mismo con el Alexa 647 y la loficocianina, que son incompatibles en convencional, y tenemos, en este caso, un 0,91 de índice de similitud. ¿Esto qué quiere decir? Que tenemos una mayor disponibilidad de fluorocromos a la hora de trabajar en un citómetro de espectro completo respecto a un convencional. Pero hay más cosas importantes en una citometría de espectro completo, y es la autofluorescencia de las células, porque fijaros en este ejemplo, estos son dos huellas espectrales de linfocitos sin teñir en un protocolo de FlowFish, que luego por la tarde habrá algo de FlowFish y telómeros, súper interesante. Y justo abajo es la huella espectral de linfocitos, pero fijados solo con un 1% de paraformaldehído. El protocolo de FlowFish lo que tiene es una fijación con formamida al 70% más una incubación a 82 grados, ¿vale? Con lo cual, esto modifica las propiedades químicas y, por lo tanto, de autofluorescencia de estas células. Y estamos hablando de linfocitos. No estamos hablando de fibroblastos o una línea celular muy autofluorescente. Estamos hablando de linfocitos que de por sí no son excesivamente autofluorescentes. Pues si comparamos, podéis ver de que hay un mayor nivel de autofluorescencia en estas células y con este protocolo de FlowFish que respecto a una fijación más estándar de 1%. Esto es una señal fluorescente más adicional a lo que tenemos en un panel de anticuerpos con fluorocromo. Es decir, la autofluorescencia es clave para muchos aspectos. Porque al final, la clave del éxito de un panel de citometría espectral está en ser capaces de diferenciar e identificar todos los parámetros, mejor dicho, todas las fluorescencias, todas las señales de luz que tenga nuestra muestra. Y esto no lo he incluido, pero imaginaros que estáis haciendo un estudio de hipersensibilidad a un fármaco. Si ese fármaco, que en algunos casos puede ser, o esa droga, produce fluorescencia, es una fluorescencia más de la que tenéis en la muestra, adicional a vuestro panel. Y es posible que os estorbe y no os permita diferenciar señales fluorescentes en determinadas longitudes de onda. En un citómetro espectral, sería coger esas células con ese fármaco, ver su patrón, su huella espectral y sustraer, porque aquí está la clave. Es decir, todo esto que os estoy explicando de las huellas necesita un paso más allá, y ahí es donde entran los algoritmos para poder diferenciar estas señales. Porque esto al final tiene un workflow que es lo que se llama el unmixing, que al final es una deconvolución, y lo habitual es utilizar, pues como si fuera un protocolo de compensación habitual en citometría convencional. Tienes un control sin teñir, unos tubos con el fluorocromo, el reactivo, el anticuerpo o el colorante individual para identificar esa huella espectral. Y con un algoritmo de mixing se van a convertir los datos de estos, en este caso, en este equipo de los 64 detectores, en parámetros de fluorescencia, que es lo que se llama los datos ya unmixados, los unmixed data. Por lo tanto, al final, la clave de todo esto es el algoritmo de unmixing, porque los citómetros de flujo, la única diferencia, un citómetro de espectro completo es un citómetro de flujo convencional con muchos detectores. Pero la clave es esta diferenciación. Esto quiere decir, y si lo aplicamos sobre todo al campo de la inmunología, claro, tenemos muchos más fluorocromos que podemos diferenciar en una misma muestra. Por lo tanto, vamos a generar datos de una mayor profundidad inmunológica, pero no solo fenotípica, sino también funcional. Claro, esto nos lleva a tener mucha más información en una misma muestra y nos permite revelar a veces patrones o tipos celulares en los cuales, bueno, suelen ser muy poco frecuentes, pero tenemos la capacidad de diferenciarlos. Nosotros cuando empezamos con la citometría espectral hace 4 años, y en un panel que luego os explicaré de 37 marcadores, te encontrabas tipos celulares que dices no puede ser que existan. ¿Por qué? Porque, bueno, pues te puedes encontrar pues, monocitos que no son HLA-DR positivos y resulta que tienen marcadores de NK. Bueno, esto cuando tienes toda esa información te permite ver patrones que generalmente no se habían abordado. Obviamente la parte de autofluorescencia es muy importante porque permite aumentar, esta sustracción permite aumentar la resolución de tus lecturas. Y otra cuestión importante, sobre todo a nivel de análisis, es que estos datos permiten lo que se llama los in-sample controls. Supongo que habéis oído hablar que en citometría de flujo, uno de los controles que más se utiliza, pues se pueden hablar de controles de isotipo, que a mí no me convencen, los famosos controles FMO de fluorescent -1, pero el mejor control de todos es que si tú tienes e identificas un tipo celular dentro de tu muestra que sabes que es negativo para ese marcador, eso es un in-sample control. Cuantos más datos tienes, obviamente te permite hacer esto. Reduce la cantidad de tubos que tienes que hacer y es más eficiente a la hora de analizar. Por otro lado, cuando se hablan de pues 40, 50, 30 y tantos marcadores a la vez, lo primero que debéis pensar es, si me cuesta a mí hacer un diseño de panel para 10 o 12, no me quiero ni imaginar lo que puede ser hacer un panel de 40 marcadores. Hay una mala costumbre en, yo me quiero comprar un equipo de citometría y habitualmente hago paneles de 8 colores. Pues me compraré un citómetro de 8 detectores de fluorescencia. ¿Para qué me voy a gastar más en uno de 12 o de 14? Eso es un error. Es decir, aprovechar al máximo la capacidad de un citómetro no quiere decir de que lo estés haciendo correctamente. Tener la flexibilidad de tener suficientes detectores como para realizar un diseño de panel lo más eficiente posible permite mejorar los resultados que se obtienen finalmente, tanto en clínica como en investigación. Por lo tanto, aquí podemos hacer muchos marcadores, con lo cual nos da flexibilidad. Y aparte, y esto ya es para los clínicos, ya está homologado como equipo de diagnóstico. Con lo cual, una de las cuestiones y que va a ocurrir en los próximos años es la entrada de esta tecnología en la clínica. Vale, y a nivel de inmuno, lo que es inmuno y sobre todo, ahora que está muy en boga, pues el tema de CAR-T. Claro, cuando alguien te empieza a preguntar, ¿puedo hacer un panel? Y y les preguntas, vale, ¿qué antígenos, qué marcadores necesitas? Te vienen con un listado que parecen los textos de papiro, que empiezan a decir, pues, yo quiero todos estos. Yo recuerdo que nosotros empezamos con el espectro completo en 2020, justo con la pandemia. Y pasamos de hacer paneles de 15, 16 marcadores a hacer paneles de 37, 40. La gente cuando pide no tiene término medio y los inmunólogos menos. Entonces, claro, ahora con todo esto, que si PD1, que si CALG1, que si TIM3, que si el CTLA4, LAC3, el 2D4, el CD57, aparte de marcadores de línea, para T-Rex, para los distintos tipos de células NAÍVE memoria, o sea, ya no me sirve lo del CD45-RA, CD45-RO. Eso es ya no del siglo pasado, sino del anterior. Entonces, ¿qué ocurre? Para poder bucear dentro de la complejidad del sistema inmune, necesitamos muchos marcadores. Claro, esta tecnología, como la que después también explicará Jaume, es ideal. ¿Por qué? Pues porque te permite tener mucha información en un solo tubo. Y entonces es cuando empieza la guerra de a ver quién hace más colores a la vez. ¿No? We will win the world record, the Guinness World Record of the colores. Con lo cual, esto es enero 2024, presentado en el Internacional de Citometría, hace poco en Edimburgo, e incluso creo que hubo un preprint el año pasado. Pues 50 colores para hacer las T y dendríticas. ¿Vale? 45 colores. Esto es un preprint, pero saldrá como OMIP de la revista Cytometry en breve. Hablando de solo para las células de memoria. Nosotros fuimos un poco más modestos y en 2021 sacamos un capítulo pues, de 37 marcadores. ¿Pero qué pasa? Eso está muy bien. Pero lo de garbage in, garbage out es clarísimo en citometría de espectro completo. Es decir, y en citometría convencional. Esto le encantará a Jordi Pérez. Los de estos. Porque, claro, hay mucha crítica a las publicaciones de distintos artículos y la calidad de ellos. Entonces, se publicó hace nada, pues este artículo es de 2020, pero un expression of opinion. ¿Por qué? Y solo quiero que miréis ahí a la derecha. O sea, uno que haya hecho citometría, cuando ve poblaciones y dispersión de ese tipo, dice, esto, con perdón, es un churro. Los mismos editores de Cytometry tuvieron, bueno, a partir de esto. Con lo que os quiero decir es que lo importante no es el número de fluorocromos y marcadores que hagas a la vez, sino la calidad de los datos que obtienes. Tanto para investigación como para clínica. Y, bueno, en citometría al final, pues tenemos muchos problemas. Lo que habéis visto antes es un problema de spread, de dispersión, y esta dispersión pues, puede venir de fluorocromos positivos que provocan en la población negativa, en otros detectores, pues dispersión. Pero en citometría de espectro completo hay spread que no está relacionado con la presencia de ningún fluorocromo. Con lo cual, esto puede sonar raro, porque esas dispersiones que habéis visto antes generalmente suelen estar provocadas por este fluorocromo con su intensidad en otro detector. Y aquí, cuando tú utilizas dos fluorocromos con un índice de similitud relativamente cercano a 1, como el que está en la diapo, el de 0,91, y miramos la dispersión. Estas son muestras sin teñir. La dispersión en el detector generado de APC, cuando tú en el experimento utilizas APC y anexo 647, aunque tu muestra la que estáis viendo es negativa, el spread que tiene es 2,5 veces mayor que si en el experimento haces una mixing de ese algoritmo solo con el APC y no utilizas la huella espectral del anexo 647. Lo que os quiero decir es que, obviamente, lo importante siempre cuando se hace un diseño es que la diferenciación de estos patrones de spread hay que tenerlos muy claros y hay que vigilar con esto. Sí, iré muy rápido ya en un poco la última parte. En 2020 nos implicamos con proyectos de profile inmunológico en COVID, después en long COVID cuando estábamos en IGTP. Después también con IrsiCaixa, la parte de long COVID, incluso también monitorización de pacientes trasplantados de riñón y ver un poco el efecto de la vacuna. Este fue el panel que se diseñó en su día, de 37 marcadores. Aquí, obviamente, la idea era pues, ver distintos tipos celulares, no solo hacer énfasis en T, sino eran Ts, Bs, NKs, monocitos, dendríticas. Obviamente, el diseño de estos paneles pues, es algo más complejo que en citometría convencional, solo por el número que tenemos. Y es muy importante la optimización de las tinciones, de los anticuerpos. No se pueden poner 37 anticuerpos a la vez, ni 40, sino hay que hacer un estudio preliminar, una optimización del protocolo. En este ejemplo, es la expresión de CXTR5 en unas condiciones de protocolo en el cual ese anticuerpo está dentro de una chemokine mix, una mix de los anticuerpos para quimiocinas, y lo extraes del step del punto de inclusión de ese anticuerpo y lo cambias a un paso anterior, pues fijaros la diferencia de la tinción de CXTR5. Es decir, todos estos protocolos con tantos anticuerpos requieren, como casi todos en citometría, pues una optimización, pero estos con tantos anticuerpos, obviamente hay que hacerlo con más cariño. Y para terminar, la clave también de todo, porque podéis generar data files de 40 marcadores, pero ¿cómo se analiza eso después? Tienes el guapo que se pone guapa que se pone a analizar esto en Canucha, en Flowyo, con lo cual podéis utilizar el método tradicional o utilizar herramientas para ello. Claro, esto es lo que salía del panel de 37 marcadores. Lo puedes hacer en una muestra o en varias para verificar qué distinciones son las correctas. Pero esto es morirse con un montón de muestras. Claro, tenemos herramientas. Lo que pasa es que todavía no estamos acostumbrados a estas herramientas. Reducción dimensional, o sea, reducimos 37 o 40 dimensiones a 2 dimensiones. Hacemos clustering, que sería similar, aunque no es exactamente igual, pero similar a lo que sería el gating manual que hacemos. Y todo esto al final ha ido evolucionando y hay varias plataformas en cloud que te permiten subir esos ficheros y poder analizarlos con distintos algoritmos. Esto al final, obviamente, requiere un proceso, eso casi daría para una charla completa. Al final, nosotros en el proyecto de COVID, en el más grande que tuvimos en 2020-2021, llegamos a procesar 476 muestras, con lo cual esto al final generamos por varios time points 2.856 ficheros de datos. Llegamos a, una vez descartado células muertas y agregados, pues 1.500 millones de eventos. Claro, esto es complicado de analizar. Seguimos una serie de procesos con un cleaning previo. Trabajamos en una plataforma, principalmente en Omicue. Y esto al final, pues bueno, obviamente te permite tener un análisis de los datos. Podríamos decir más objetivo, bueno, hay a veces dudas de cómo configurar esos algoritmos, ¿no? Pero esto es una de las cosas que van a tener que ir evolucionando en los próximos años a nivel de la citometría computacional. Y para terminar, al final, la citometría de efecto completo nos va a permitir, sobre todo, generar unos datos de alta dimensionalidad, que nos va a permitir también, sobre todo con la extracción de la autofluorescencia, ganar en resolución. Obviamente van a salir protocolos y están saliendo protocolos que no es hacer muchos marcadores. Con lo cual, al final también tenemos algunos protocolos pues para todo lo que es partícula pequeña, que también se pueden utilizar con citometría espectral. Una de las ventajas es que esta tecnología obviamente te permite hacer sorting, o sea, separar, tener un citómetro de espectro completo, separador, y te permite aislar las células en función de los parámetros que hemos elegido en el PPQ. Y ya está. Gracias y que la fuerza espectral los acompañe.
Si hay alguna pregunta, la podéis hacer ahora y al final dedicaremos un poco de tiempo a la puesta en común. Si alguien tiene alguna pregunta.
Hola, dos preguntas. Y primero, a la hora de diseñar un panel, ¿cuánto es importante el complexity index del panel mismo? Cuando se va a diseñar un panel con muchos colores. Y la segunda pregunta, claro, tendríamos que marcar, ¿no? por tanda, o sea, en un panel de tantos colores tipo 36, 37, ¿cuántas tandas hay que hacer? ¿Y cuánto, con qué orden, en qué sentido? O sea, primero lo que tiene baja expresión y luego subpopulaciones, o más bien nivel de expresión de antígeno.
Empiezo con la segunda. Las tandas. Las tandas al final están pensadas, o sea, tú partes de una bibliografía previa, obviamente, y una de las cosas que dice la bibliografía es que los chemical receptors se ganan en resolución cuando haces una tinción secuencial. Entonces, eso lo comprobamos y realmente es así. Y entonces nosotros siempre marcamos inicialmente, o sea, primero hacemos viabilidad eliminamos la viabilidad y después ya empezamos un secuencial. Entonces, PCR7 y CXTR5 nos funcionan muy bien al principio. Después una CYTOMICS, porque para subtipos de célula T, pues CXTR4, CXTR3, CXTR6, CXTR7, hay un montón. Te das cuenta que si eso lo pones con la mix general, baja mucho la expresión. Lo que tú tienes que hacer es un análisis inicial de comparar la tinción de un marcador cuando está solo respecto a cuando está con todos. Entonces, cuando pones tantos anticuerpos a la vez, siempre hay interacciones y la cuestión es identificar esos problemas para sacarlo de alguna manera de él. Entonces, nosotros habitualmente, pero depende de los antígenos, hacemos, aparte de la viabilidad, hacemos 3 tandas. De la primera se me ha olvidado.
Sí, era sobre complexity index del
Vale. A ver. Ahora estábamos diseñando un panel de 45 y el complexity index que nos sale es la mitad del que teníamos en el de 37. Has aumentado, pero... A ver, es un dato importante, pero yo me fijo más en la similarity. Es decir, en cada pareja, cuál es, pero intento, porque me he encontrado que en distintas casas comerciales tienen alguna, en su web, alguna aplicación, y no coinciden los similarity index. Lo ideal es intentar probarlo. Claro, si no quieres comprar el anticuerpo, pero ayuda mucho, casi todo lo que esté por debajo de 0,9 no suele dar problemas, pero tienes que vigilar la expresión antigénica cuando tú estás poniendo 2 cromos muy similares, ¿vale? Que sean excluyentes entre ellos o que realmente sea un antígeno primario, ¿no? El famoso artículo de Mario Rodríguez, primario, secundario, terciario, o sea, que se exprese como un CD4, un CD3. Entonces, tienes que jugar con eso. Y después tienes que jugar con la disponibilidad de los protocolo de las casas comerciales, con lo cual es un problema. Pero tienes que tener en cuenta esto. A mí me gusta más mirar la similarity que no la complexity.
Bueno, muchas gracias por la charla. Yo tenía una duda. Si tú diseñas un panel, pasas tu reference control y haces el mix y otro día pasas una muestra que te falta un reference control, ¿puedes utilizar la misma mix o tienes que volver a pasar todo el reference control sin el que...
Puedes utilizarla. Sí. Tú puedes. Es decir, estos equipos generan dos tipos de ficheros. Lo que se llama el raw data, los datos crudos, que son las señales fluorescentes de cada uno de los detectores. Y después está el AMBEX. Con lo cual, si tú, al día siguiente, al cabo de la semana, te añades el nuevo, raw data, haces el unmixing otra vez y entonces se lo... Claro, es uno de los problemas, ¿no?
A ver, es un tema sobre, igual que has dicho que hay una carrera, a ver quién es capaz de poner más por ejemplo, los cromos en un panel. También hay una carrera por parte de las casas a ver quién tiene el equipo con más detectores. Hemos pasado de 64 a 200 equipos. Yo tengo la sensación de que a nivel de tener muchos más canales, no nos va a permitir tener muchos más marcadores, sobre todo por el tema que hablabas del spread vinculado al mix. Pero hay un punto que es el tema de las autofluorescencias, ya no como un problema que tienes que quitarte, sino las autofluorescencias como algo que puedes utilizar como una marca propia de una célula, que está muy bien descrito en los eosinófilos, pero yo creo que en otras poblaciones puede ser de interés. En este caso, por ejemplo, el tener un equipo de 200 o 200 canales, pues puede dar una ventaja frente a tener un equipo con 64 canales. Y lo digo porque en general las autofluorescencias son diferentes, pero se parecen mucho entre ellas, mucho más de lo que se parece un fluorocromo a otro.
Sí, claro, aquí hay un problema. Es decir, para generar las huellas espectrales, cuantos más detectores tengas, mejor. Entonces, hay un equipo, por ejemplo, que tiene un montón. Pero tiene un montón de 500 nanómetros en adelante. De 500 hacia abajo, no. Con lo cual ahí se te ha complicado un poco. Pero después, los valores de área, altura y anchura que tú puedas sacar de intensidad de fluorescencia no es la suma de esa huella espectral, es el detector máximo. Con lo cual, si tú lo reduces mucho, vas a reducir la cantidad de fotones que tú vas a poder detectar. Entonces, hay que llegar a un compromiso de poder resolver distintos fluorocromos con el número de detectores. A mí me gustaría hacer, porque hay algunos estudios, pero a mí me gustaría hacerlo. Tenemos y con otros, porque en el site de Edimburgo creo que ya han salido 2 especiales más. Ahora ya tenemos como 5. Pues comparar la resolución, pero es que la resolución también depende de muchas cosas. Si el detector es un fotomultiplicador, es una PD, el ruido electrónico de fondo, o sea, es una serie de sumas. Pero cuantos más mejor hasta cierto punto.
Pregunta muy simple. ¿Qué algoritmo recomiendas para analizar los datos? ¿Cuál es más intuitivo?
Intuitivos no son ninguno. Es decir, a los citometristas tradicionales están analizando en Floyo, en Viva, Calucha. Cuando te pones aquí tienes que cambiar el chip absolutamente. Entonces, entras en la reducción dimensional. Hubo una época que Disney era el que más o menos se utilizaba más. Ahora parece ser que el Yuma será mejor la estructura global y después, pero la reducción dimensional no es más que una puesta en escena gráfica de lo que tú tienes con muchas imágenes. La clave es la clusterización, que es como si fuéramos. Entonces, claro, ahí tienes Flowzone, tienes Xenograph, van saliendo más, hay limitaciones. O sea, a mí personalmente Flowzone es el que de momento me da más comodidad a la hora de analizar los datos, como algoritmo de clusterización, pero esto va muy rápido. Pero tienes limitaciones porque no puedes poner todos los eventos, tienes que también coger una parte, porque no hay la capacidad, no tiene la capacidad para manejar tantos eventos, puede manejar hasta por 4. Nos falta, es decir, la figura del bioinformático citométrico es algo que ahora mismo es indispensable y no me sirve un bioinformático genómico. Tiene que entender lo que es la citometría. Y esto ahora mismo está muy buscado.
Sí.
Bueno.
Las tres. Pasemos. Our next speaker is Dr. Jordi Pérez, well-known researcher, an expert in functional cytometry. So he's going to talk about live cell mapping, analysis, and acoustic. ¿No hay una carpeta que se diu Immunologia?
Okay, maybe inside this folder. This one, I think. CI. Yes. Okay. Thank you for inviting me. The topic of my talk will be very different. I'm collaborating with the inventors of acoustic cytometry, and now we are maintaining, or 1 year ago, a new cytometer, a conventional cytometer using acoustic focusing, and taking advantage of this tight focusing. The system is able to catch a lot of images, up to 6,000 images per second. It's conventional flow cytometry and bright field imaging. In the future, they will use fluorescence markers, etc. This is a CYTOMICS, is a very compact cytometer, which is like the Attune NXT, the predecessor of this technology. In fact, in 2018, we published a paper about the acoustophoretic orientation of the erythrocytes, and we discovered for the first time different populations of different clusters of erythrocytes. Erythrocytes in a conventional cytometer are a common cluster. And by using this kind of cytometers, we will be able to discriminate different subpopulations. They decided, the inventor of the acoustic cytometer decided to add a objective they buy a very expensive camera for that. And based on this basic experiments, they developed this cytometer. In fact, our cytometer is equipped with 4 lasers, and it can combine up to 14 color flow cytometry. And the resolution is very good here. This is because it uses based on the characteristics of the optics. This specific cytometer is not designed to measure nanoparticles, but you can detect nanoparticles as we have measured on this flow cytometer. In fact, this is the conventional cytometry using a objective, and then based on the third, will catch these images on the flow cytometer, up to 6,000 images per second. And this is just to show you the advantage of the. It's working here. This is to show you the animation. You can unplug the acoustic focusing on the cytometer, and then this is with the acoustic focusing off, and it's very difficult to focus this particle. But using the acoustics, you are able to have a tight alignment of the cells, and then this allows. This is the main advantage, to catch a lot of images per second using this kind of cytometer. Now we are using this methodology to investigate. I was fascinated for the first time. My first weeks using the cytometer were only dedicated to try to understand why I was looking for in the cytometer. Now this is a conventional. This is only a forward versus side scatter parameters, and then you can gate your cells and then obtain the images. During the acquisition, you can perform a fine focusing depending on the size of the cells, and also by gating the population of interest, because based on the size and the scatter properties of the different populations, you will have a different focusing. Also, you can acquire the cells at a high sample rate, but you will need to adjust this fine focusing using the panel, which is quite simple, only changing these parameters. The main problem is that the FCS file will be larger in comparison with conventional FCS file data. And now the modern software, because this is generating, the software is generating a different parameters that can be combined to the conventional flow cytometry, because you can measure different parameters, morphological parameters related, such as this one, to the area in microns, perimeter in microns, etc., and a large of parameters that you can combine with the conventional information using flow cytometry. This is the classical immunophenotyping, as you will see. And also about the complexity of the cells, just to distinguish healthy cells from sick or dying cells, based on the entropy intensity. This is the pixel intensity that is registered on the different images, and then you can combine these parameters to also discriminate and visualize the cells in your cytometer. This is just most of the experiments I will show you today are done with maybe 1 color or are 5 colors, not more. But this takes advantage because you can characterize your cells and quickly identify the dots here. This is just to show you the lymphocytes, the monocytes, the neutrophils, and the eosinophils, as you can see here. This the hematologists are shocked because, and maybe the immunologists too, because the backfield imaging is completely different to the conventional smears used for microscopy analysis. There's a McRumbel, GEMSA, etc. And now we are working on in a potential atlas to compare pathology samples with conventional optical microscopy and the images that we are obtaining using conventional cytometry. This is a magnification. This is a resolution provided by the lymphocyte monocytes, neutrophils, and eosinophils, as you can see here. And these are megakaryocytes, can quickly identify it using the flow cytometer. In addition, this is very good to discriminate potential artifacts, and you will see now really interesting information about a protocol which is very conventional about the identification of singlets and to discriminate. Okay. Now, again, the lymphocytes, monocytes, and neutrophils compared with plasma cells, as you can see here. This was done by performing immunophenotyping and then gating the CD138 population and to visualize the plasma cells here. And this is related to the topic discrimination. This was on the topic there that Marco and me were discussing during the first days using the cytometer. And now you can see here, because the system is also analyzing the cells that are, the images that are correctly taken on the flow cytometer. And then because the cells normally are in the center of the images, and then you select the images that are well selected. And this is a staining, no, this is a conventional approach to discriminate erythrocytes using the forward scatter with a height parameter and the area. And now here I'm selecting this region. You will see later the information with this region. But as you can see here, I can plot this gate and discriminate 3 different populations using the imaging information collected by the cytometer. Here we have only this population, in principle, doublets and aggregates, and here we can discriminate these 3 different populations. Okay, we'll see what happens. This is the region 6, and we show that we have a lot of singlets, not only doublets. We have doublets and a lot of coincident events. This is crucial because if we are performing a cell immunophenotyping, we need to know that in this region we can have a lot of singlets. In addition, if you have a cell sorter and you discriminate aggregates, you can deplete your sample because a lot of singlets will be included in this region. By using the different gates, using the morphology parameter, I can select the singlets, as you can see here, the true singlets contained within the region R6. Now I'm using R10 here, and I'm identifying only the singlets. These are doublets in region 7, and also coincident events here. And that's very nice because now we know that we have a problem in discriminating singlets, and we can easily visualize the problem as you can see here. Also, in my lab, as you said, we are performing functional flow cytometry, and we are combining classical immunophenotyping with enzyme function, etc., which is crucial to identify very rare cells such as cancer stem cells, etc. And we are using a minimal sample perturbation approach, which is not new. This was published in Cytometry Part A in 1998, showing how using the violet side scatter, and in this case the red side scatter, we can discriminate platelets, erythrocytes, granulocytes, monocytes, and lymphocytes. On the side peaks, you can also discriminate the same by combining even the violet side scatter, in this case with the blue, but you can use the green, yellow laser or the red cell laser to discriminate these different populations. And now we always avoid, if we stain the marrow or we stain the blood, we directly take an aliquot of this marrow specimen, and then we add the antibodies, the substrates for the determination of potential membrane activity or enzyme activity. And then by simply diluting the sample without performing an enrichment of the cells or the discarding red cell lysing solutions, we can discriminate platelets, lymphocytes, monocytes, granulocytes, and red blood cells, as you can see here. This is simply by changing only 2 filters on the flow cytometer. and changing this back, and then you can easily collect the violet side scatter to discriminate these cells in blood. This is crucial to identify populations that can be very sensitive for the lysing solution, such as the precursors of our red blood cells. And here we have discriminated the erythrocytes and the platelets, and we have only lymphocytes, monocytes, and neutrophils here, and I've selected these two regions. As you will see, we can easily visualize the precursors of the erythrocytes, which are mainly erythroblasts in this region. This was R4, and this one, and a lot of erythrocytes, which are coincident with the cells because we have avoided the lysing solutions. So we can also visualize the erythrocytes here. This is crucial to study also leukemias which are derived or originated in the red blood cell compartment, such as in myelodysplastic syndromes, etc. And this is specifically for you because this is a combination for the different markers to discriminate plasmacytoid dendritic cells, conventional dendritic cells, and basophils. And you can see here the plasmacytoid dendritic cells, conventional dendritic cells, and basophils, because you can plot and gate these cells and visualize this region. In fact, you can only by selecting the cells, you can identify the cell of interest. Or on the contrary, you can backgate this image and plot on the cytometer where this cell is located. Of course, we are using, and you mentioned a very important topic in your previous presentation about the fixatives, etc., because this alters the autofluorescence, etc., but also alters the morphology of the cell. This is a no-wash protocol, which is conventionally used in many laboratories all around the world. And this was done using a dendritic cell leukemia. As you can see here, I've selected the positive population of these dendritic cells, and the following slide is the same sample. This sample was prepared with the minimal sample perturbation protocols using the no-lyse, no-wash approach. And as you can see here, we can easily identify these protrusions in this dendritic cell leukemia, which is not possible to see here because you have only changes, important changes in the morphology of the cells. And also, if you deplete these dendritic cells, the shrinking will also affect the intensity of the fluorescence. So we always prefer to use these kind of protocols to identify very rare populations. CAR-T, you mentioned also CAR-T, and cytotoxic activity can be easily monitored and using this cytometer. Of course, using a conventional cytometer, you will be able to measure the cytotoxic activity. We have published different papers on this topic, but you can easily identify how the lymphocytes, the cytotoxic cells are interacting with tumor cells or with leukemic cells to try to trigger the cell death. And as you can see here, this is an interaction with this cell, which is a cytotoxic cell with a tumor cell, now inducing some blebs at the end, provoking the cell death of the tumor. You can easily visualize even few dots on using this cytometer, and also confirm the true nature of the cells, the dots that can be apparently dimly expressed markers or other populations that you can have the suspicion that this can be an artifact or not. No? Acute leukemia. This is just to show you acute leukemia cells, and this cytometer also takes advantage because you can easily identify acute myeloid leukemia cells because this is a specific presence of our rods, as you can see here, and you can easily distinguish, even in the absence of markers, the presence of acute myeloid leukemia cells, and this hallmark, which is characteristic in AML, the presence of these our rods. In addition, by combining the forward scatter, in this case, only one marker, a leukemia with increased expression of CD34, you can visualize the presence of small CD34 positive cells and the presence of large CD34 positive cells, which are leukemic, of course. And also to discriminate human leukemia and healthy cells. This is a case of a human leukemia. You can see a abnormal distribution. You can see the classical pattern of lymphocytes, monocytes, or neutrophils based on this invasion of leukemic cells. These cells were stained with DAPI. We commonly use DAPI to study also the cell cycle and ploidy in human leukemia. And in addition, we added a marker because this population, this leukemia had a dim expression of CD117, as you can see here, and bright cells here. Now, using the images that were well processed and it was centered on the image, we were able to identify also, because you can add the particle count, which take advantage to identify very tiny particles, such as liposomes, you will see later, discriminate different populations. This is the A parameter obtained using this morphology analysis and the combination with the fluorescence parameter. And at the end, identify healthy cells and human leukemic cells in the same specimen. Also compare the difference in the DNA. This is the DNA from the healthy cells. This is the DNA content of the abnormal cells, the leukemic cells. And this is the overlay of these two plots showing that the leukemic cells have an increased DNA content, which is a near diploid population as confirmed in this overlay. And just to show you the populations in the normal cells here, the healthy cells here, and the leukemic cells here. You can see the abnormal cells are larger in comparison with the healthy cells. Also for MDS, you can also discriminate. This is a conventional staining optical microscopy analysis of the cell. And this is a mitotic catastrophe phenomenon that occurs in MDS, myelodysplastic syndromes. Okay, and this is crucial because this can be a red cell tragedy and increased risk of a relapse of these patients with MDS. And easily visualize by staining the DNA these multinucleated populations and using this method to confirm that there are a lot of cells in this case with this aberrant mitosis, and this can be very informative for the clinicians because this is an increased risk of progression of the MDS to human leukemia. And this is the same patient that was stained with Mayrumbal Yemsa, just to show the presence of this multinucleated malignant cell smear. Also for the identification of tumor circulating cells. This is the migratory melanoma metastatic cells that can be easily identified on peripheral blood. As you can see here, I will go back to show you the normal cells in blood, which are circular. But when we observe in peripheral blood this kind of oval nuclei-shaped cells, we can confirm that there are melanoma cells migrating to blood and circulating, and easily identify it, even you have only a few dots on your flow cytometer. This is a very nice example of how we can identify apoptotic cells, in this case apoptotic bodies, as you can see here, and apoptotic cells within this region, without staining, only confirming by the scatter properties, and then you confirm that you have here a lot of apoptotic cells and apoptotic events, and also to measure the chromatin condensation. And again, as you can see here, the cells in A and B and C, this is this region magnified, involving the classical chromatin condensation pattern, as you can see in these different images. Also, for the cell cycle, to visualize the presence of cells in the G0, G1 phase, S phase, and G2M. And this is very nice because we have also a large interest in the classification of the erythrocytes. The acoustic focusing takes advantage because you can clusterize different populations of red blood cells. And this was done simply by diluting 2 microliters of blood in Hanks. We always use Hanks balanced salt solution in my lab instead of PBS. We can discuss this later. To show abnormal red blood cells. This is only 1 minute on the flow cytometer, and then you can easily identify the abnormal red blood cells. As you can see here, this is a case of poikilocytosis. And also you can take advantage because you can measure yourself. This is a healthy donor. The area of these erythrocytes is commonly around 80 micron size. And this is a comparison with a PNH. And as you can see here, these large erythrocytes, which are characteristics of PNH. Also, and this is shocking because we are also interested in MRD and measurable residual disease, and we are comparing the results obtained in a clinical hematology laboratory using a special cytometer marketed for CIVD. And I was shocked again, the staining with DAPI, because you also can confirm the presence of cells with a change in the DNA content. So this can help very much to identify malignant cells. In this case, this is the distribution of lymphocytes, monocytes, and neutrophils, the eosinophils, as you can see here. And by adding the CD138 to detect malignant plasma cells, which commonly are very bright in multiple myeloma, I was detecting this rare population, which is around the 0.08% of the cells. And you can take advantage looking at this population, confirming the true nature of malignant cells, because they have, as you can see here, Dutcher bodies. The presence of these Dutcher bodies are quite different to conventional imaging, conventional microscopy analysis, and appears darker using this bright field imaging cytometry. So we are interested in perform this kind of atlas that I will hope to publish shortly, in addition to this information I'm showing today. Also for the detection of small particles. Using conventional flow cytometry, you cannot discriminate aggregates and singlets. So the image here take advantage. You can classify, you can add the particle count on your flow cytometer. And also because based on the identification of single liposomes and aggregates, etc. So again, we are classifying the images that are well processed here, and then looking at the particle count based on the image on the system. So we can compare events and all events in the flow cytometer about this population of neutrophils. Liposomes. And then look at the cargo, because you are adding, if you are adding a fluorophore and you are engaging a fluorophore inside the liposomes, you can also monitor the difference in the cargo of this fluorophore, as you can see here. And I'm going to finish my talk because I'm trying to run a lot of different specimens in the flow cytometer. This was done in collaboration with the University of A Coruña to study microalgae on the Attune CYTOMICS, to look at the different populations, the different clusters based on the emission of these fluorophores naturally contained in these cells, and looking at the different clusters based on the red fluorescence and the yellow green fluorescence of these cells, and identify different subpopulations, different populations of microalgae present in the region in R7, in R8, R9, and R10, easily classifying these microalgae, identifying the different species contained in this sample. In summary, best to say that the CYTOMICS is a conventional flow cytometer equipped with this high-speed bright field camera, and you can automatically analyze your cells, even stained or not stained. I believe that in the future the flow cytometers will be equipped, most of them, if not all, with cameras in this way, or maybe evolving. In this year at the CYTO, it was introduced a new flow cytometer marketed by MIFTE that will have around 400 detectors, which is crazy. Okay, and just to finish, to thank the people, the CYTOMICS team, especially Michael DeWard, which is one of the 3 inventors of the acoustic focusing cytometry, the CYTOMICS team. We are improving the system and the software analysis of this cytometer, and also the people in the IGDP, because we are collaborating in many different fields. Ángel Estit from the Universidad de Coruña, and thank you so much. That's all. I will be very happy to receive questions from the audience. Is believing. Yeah.
We are running late, so let's keep all the questions at the end, if it's okay for you. And our next speaker is Jaume Coma, from the head of the unit of cytometry on the campus. Diagonal campus. And he's going to talk about cytometry.
Okay, thanks to the organization for giving me the opportunity to talk about mass cytometry today. Yeah, of course, mass cytometry is a part of a technique inside all the possible cytometry. Cytometry means measuring of cells. So if you are dealing with a short number of cells, but you need a very high detail, you can access to all the microscopic techniques, right? Well, on fluorescence, confocal, or electronic, whatever. But if you are dealing with populations with a lot of cells, you have to go to the flow cytometry that has been working for more than 50 years. And the 2 main characteristics on flow cytometry are, first, the speed. You are measuring thousands of cells per second on one hand. On the other hand, the multiparametric analysis. You can stain your samples with a lot of different markers, so you can make a deep analysis on your cells. In the last 10, 15 years, there are new developments on flow cytometry. As it has been told, spectral flow cytometry allows you to incorporate more and more colors to your analysis. But on the other hand, what I'm trying to present today is the mass cytometry, the site of analysis. And from the beginning of the history of flow cytometry, there is a race in having more and more colors. Sometimes maybe it's not very useful, but we are always to obtain instruments that can detect more markers, because we want to analyze complex systems. And the immune system is the typical complex system. Keep in mind that if you want to make a deep analysis on T lymphocyte subpopulations, you need about 25 different antibodies. So you need instruments that can face this kind of analysis. And that's why typical flow cytometry has incorporated more lasers, more detectors, or even more capabilities. But the problem of dealing with fluorescence is that we are in a short part of the electromagnetic spectrum. We're dealing with visible light. So you're moving from, let's say, about 400 to 800 nanometers. This is a very narrow part of the total spectrum. And if you're looking on the fluorescence emission of your fluorochromes, you will see that they are quite wide, and they tend to overlap one to the other. So it's a problem when we want to make a lot of different stains. So that's why flow cytometry always ask you to bring us controls. So this is a control non-stained sample, because we need to measure the autofluorescence, but also we need the single stain controls to calculate the complex compensation matrix or to take a look on the spectral fingerprint if you are using the full spectral cytometry, right? And from here, you say, okay, but what happens if we obtain something like this in our analysis? Look at this really sharp and very clean peaks. That's what mass spectroscopy provides us when doing an analysis, because in mass spectroscopy, the resolution of the different peaks is really, really nice. So someone has a nice idea to have, okay, what about doing, making a flow cytometer that instead of working with fluorescence, we are labeling our antibodies, our markers with methyls, and we use a mass spectroscopy detector to detect our cells. And that's what mass cytometry, CYTOF, is doing actually. In principle, our cells should be stained with antibodies, but no more fluorescence. These antibodies are attached to methyls, isotopes of methyls, right, are introduced inside of our instrument. There is an nebulization, there is a cleaning of the low weight molecular elements, and then with the time of flight, that's why we call the CYTOF time of flight system, are injected and detected in the basis of cell per cell, right? In fact, the resolution is so good in this kind of systems that we can even detect or difference in just one atomic mass unit from different isotopes. So this means that we can use, let us say, the 3 isotopes of erbium that are only one atomic mass unit difference between each other to attach them to 3 different antibodies and distinguish them with no problems. In fact, in our systems, we have about 135 channels for detection that can range. We can detect elements from 75 to more than 200 units. So mass cytometry allows us the study of complex biological systems and a single cell level, because we're injecting the cells and detecting all the signals from cell to cell, right? As I said, cellular targets are labeled with metal antibodies, and then we can detect with time of flight mass spectroscopy. The consequence of this, using this technology, is first of all, there is a minimal background. We don't expect to find any methyl in our cells. So there is no autofluorescence, nothing about fluorescence anymore, but no background because we don't expect to find any methyl. On the other hand, as long as we have seen the peaks are really sharp and they do not overlap one to the other, we don't need to calculate compensations. So what we're going to see here is that we need no more the single stain controls. As I said, the advantages of this technology is we don't need to bring the different controls of blank samples, of single stain samples. We can analyze directly on our sample. On the other hand, as all the peaks are clearly distinguished, we can just change one tag with another one with no problems of redefining our panel, yeah? Because you simply tell the instrument which is the channel where you're going to find your methyl. So this is a very nice technique when we need to face the analysis of systems that we do not know anything about them, right? It's very flexible. On the other hand, not everything is nice in the technology. So the speed we're used to analyze thousands of cells per second in fluorescence flow cytometry. Here, the speed is about hundreds of cells per second, 500, 600 cells per second. And this means that if you want to analyze, let's say, about 40 or 50 different stains, what you have to do here is to analyze a lot of cells, and this means time. Yeah. Well, the typical time for analysis, one sample is about half an hour to 1 hour, right? On the other hand, I said that we have 175 channels, but the current chemical methods limits the use to about 50 stains, right? And of course, as long as these cells are introduced into the system and burned, we are not going to sort, never we are going to sort these cells because they are completely destroyed. But First step, you need your antibodies. You cannot use your fluorescence antibodies anymore, and you can make your own antibodies tagged to methyls. There are conjugation kits to do so, but you can purchase this. There are several suppliers of antibodies attached to methyls for CTs, for cytokines, for transcription, cell cycle, and whatever. But maybe you are interested, if you are especially for clinical analysis, in use pre-designed panels. Let's say for T-cell immuno-oncology or acute myeloid leukemia, there are combinations of different antibodies attached to different methyls, so you can directly use this combination in your system. On the other hand, you can buy pre-designed kits that not only the antibodies with the methyls attached, but also all the reagents. One thing about the sensitivity of the technique of the spectroscopy that is very sensitive to the contaminations. We are using water that has something about a little bit of lead, you will detect it. If you manipulate your sample with rubber gloves, you will have a lot of barium. So be aware that this is a very sensitive technique, that contamination is always found, right? If there is any. And now, talking about the kits in human samples, there we have been dealing a lot with the direct immunoprofiling system, which means we have these kind of kits that are 30 different antibodies right at the bottom of a tube. You just add a little bit of PBMCs or blood directly here. There are some washes and some fixation steps, but then you can keep this sample. You bring this sample to the laboratory where the mass cytometry is located, and there is a direct analysis that can allow you to identify up to 37 different immune cell populations. The nice thing is that we have a software that is dedicated to this kit, and the analysis is really, really fast, right? In fact, the stain and fixation steps can be done in the hospital or wherever the sample is taken. The acquisition with the alias mass cytometer is about 30-45 minutes, and the analysis with this software is quite direct. So it's a very fast and nice thing to do. But more than this, as I said, there are a lot of different gaps here that you can fill. You can use your direct immunoprofiling panel as a backbone, and then you can fill these gaps with your own antibodies, or use what they call expansion panels to make a deeper analysis on B cell populations, NKs, T cells, whatever, right? But the nice thing that I want to tell you today is that when you have a panel, you can add or remove antibodies with not a big problem, because you don't have difference in the staining index. You don't have overlaps from one signal to the other. That's the good thing about mass cytometry, right? More than this, as long as we have a lot of gaps, channels to open, still open, we can do the barcoding. Combining palladium isotopes or cadmium isotopes, we can stain our different samples at the beginning, and then we mix these samples all together, and then we process the rest of the staining, the injection, the analysis of the sample, and then is in the analysis step that we just recover the original procedence of these cells, right? So the nice thing of this barcoding is that you can combine these samples, and then this improves the quality, because all the staining, all the processes, and all the acquisition are done at the same time on all your samples. So it means that you are eliminating sample-specific staining and data collection variation. And it has been said before, but I like to insist in this, not only for mass cytometry, but in general, all the cytometric techniques that we are combining a lot of different stains, we have a problem with the analysis of the data. We have one color, we say, okay, positive and negative, very easy, 2 colors, 4 populations, nice, no problem. We have 40 we call or 30, 40 markers, of course, the number of combinations is so high that we can go no more with the manual gating and manual analysis strategies. So what we have to do is to use these kind of programs that has been told before about the reduction of the dimensions and based on analysis of principal components and automatic clusterization. So at the end, we have something that we can try to understand. But as it has been said with Marco, this maybe is a complete talk to be a little bit, to be better. Just an example of how mass cytometry has been used at the beginning of the COVID-19 disease. There was one of the first papers that were published. It was this one. Cited here is from 2020. It was the characterization of the first cases known it was done in Paris, yeah, by the Torre lab, and was using the direct immunoprofiling kit with a couple of more antibodies, and what they discovered or what they detected was the differences in the immune response on the people that was affected by COVID-19. The nice thing about this paper, it was done in a very, very short time. Yeah, from the acquisition of the data to the publication were only a few weeks, right? Was something that was done in a very short time because this flexibility of using a kit to do a fast analysis, and was done in a I said, well, I think about a couple of months or so. And as long as in our facility we have both full spectrum analysis and mass cytometry, some people is coming to our lab and say, okay, which is the best technology that I can use with my samples? And I always say, okay, depends on what you want and what you want to see. On the other hand, they were talking about a lot of different markers that we can analyze. We're talking about 50 in the mass cytometry, in the Aurora 40, but it's been said there are published panels about 50. So this is not very different. The speed is clearly better in fluorescence cytometry than in mass cytometry. But on the other hand, as I said, the panel flexibility, when you have a panel in fluorescence that you have been validating, you cannot change one color for another. Yeah, you have to be very critical, very strict, repeating again your panel and not changing anything. On the other hand, on mass cytometry, you can change one methyl for another antibody with another methyl, but there is not a problem. Just open the channel for collection. Yeah. So in mass cytometry, this capability of processing different samples, I didn't say these samples are fixed. You can just frozen them, accumulate them, and then send all them all together to the lab. So it's not a very big problem if you have samples stored even for months. Yeah. On the other hand, you have a lot of antibodies in your fridge that maybe you want to use. These antibodies with fluorescence can no more be used in mass cytometry. You have to purchase new or make your own by yourself. Sometimes I like to point to be aware of using your antibodies with your fluorescence directly in full spectrum cytometry. There is a lot of work previously to really validate, to adjust the concentration, to make the correct titration of your antibodies, especially when you are facing complex panels, right? And of course, you can do sorting with fluorescence cytometry. Not here, as I said, the sample was completely burnt, but there is something that you can do in mass cytometry, that's access to the image mass cytometry. As I know, this evening there will be a talk about Gloria Martus is going to talk to you about the capabilities of image mass cytometry. To just introduce a little bit, what we have is a module, the Hyperion, that is attached to the Helios, the mass cytometer. Oh, sorry, to the mass cytometer. And then this model is an image model that where you work on slides. You can stain your samples on a slide, but not with 3, 4, 5 fluorescents. You can stain up to 37 different antibodies on your tissue. And then you keep the morphological information on the tissue as long as the depth of the analysis of all these markers, right? But I say this will be developed this evening with Gloria. And just to tell you that as long as we're a facility, we offer a service for both internal researchers, but also from people from outside. Our ILIOS is a result of a collaboration of the IRB, the Institut de Recerca Biomèdica, and the University of Barcelona, if you're ever interested in the technology. And just that's all that I want to tell you. Just tell that in our facility, we have a lot of experience with flow cytometry, both conventional, we can offer also spectral flow cytometry, mass cytometry, sorting. I like to point that we have incorporated 1 month ago a new sorter that incorporates also image. But as Jordi said, I am also a believer that image will be the future of flow cytometry and will be incorporated. And also different techniques that you can also master that will be presented today that are present in our facility. And that's all. Thank you very much.
Is there any question?
Oh, because maybe.
All the speakers and afterwards we can interact.
I have for you a very short, it's a technical question. What about the titration of the antibodies? Is that an issue with this technique that you presented, or
you have to use the correct concentrations on your antibodies? But if you're using this kind of pre-designed panels or these kits, these are just to the optimal concentrations.
I thought it was some.
Yeah, this is an open question. And it's a fake question because it's more for the audience. Do you have the sensation that we are running too fast? Because for cytometries, all this advancement that we are achieving is very challenging. But the people that it's coming to the facilities, they are still having the same background, the same cytometric background than for 20 years ago. And they find these new techniques and these new challenges, and it's, I don't know, how do you feel about it?
You mean about education?
Yeah.
That we are too fast now, but definitely in the last 20 years, we don't have this revolution of the I thought it's the beginning, after that, full spectrum, so but we did a lot of education for the people, people that is working in the lab. Yes, this is a problem. We need to improve this.
I would like to say I'm involved in educational activities at the ICCS, the International Clinical Cytometry Society, and this is a big problem. Of course, I'm also complaining about the evolution of the cytomeetings, which are more technical. We have a lot of technical information, more and more technical information, and less science. This is a big problem. We are invited, overwhelmed by technologies, and we can do a lot of things using one single color. A lot of different experiments, and the people is very happy performing complex immunophenotyping, but this adds a lot of problems that they said because of the problem of the classical bioinformatic approach. Bioinformatics and genomics, we have tried to collaborate with the supercomputing center in Barcelona, and the bioinformatics, they don't have idea. They don't understand the structure of the conventional FCS file. And this is a big problem. We need new bioinformatics with knowledge in flow cytometry. And also, if you are going to the clinics, this will increase the complexity of the data.
On the other hand, I think that in the last 50 years, since 10 years ago, okay, I agree that the people that was doing 10 colors was okay, it was something amazing. In this moment, we have done a real big jump to more and more complexity. And of course, maybe the question is this complexity really needed always? Yeah, probably not. Yeah, but that's true that that if you have the tools, more people is using these tools, right? And then what we're facing nowadays that slowly, but people is using the capabilities of the technology nowadays, right?
Yeah, I have a question that is into question that I see that people now want results very fast, you know, for everything. And I think that working with a panel of 50 parameters or something like this, maybe it takes a lot of time to set up, and you have to invest a lot of time, no? But process, they want fast result, no? So I don't know how much time you need to put a 57 or 50 color panel, just to be able to avoid artifacts. But I think that it has to be a lot of time, no? So what do you think about it?
When you have done different panels, maybe in 15 minutes you finish that. So, yes, you need to spend a lot of time, but you need to go to a course activity. The exception is because people are trained for this and can help you. This is very different to go to. You can also go to the manufacturer, and different manufacturers can show you or help you to set up a panel. You have the pressure of your boss, maybe that, yeah, I need this panel, read it in 15 2 weeks. So this is impossible. So you need to realize that when you that an experiment for flow cytometry, even though it's high-dimensional or less dimensional, you need to spend some time to check your antibodies, to listen. This is very important. So because I saw a lot of data. Final of one project that is very, very complicated to analyze because the problem was at the beginning. So if you spend time at the beginning, this gives proof of success for the final for the so the time with them.
Yeah.
Marco, mira, quería preguntar. Cuando antes estaban hablando, ella quiere tirar más canales de captación en el full spectrum. Cuando luego hablas con las casas comerciales, o no las casas comerciales de los productos, sino CYTOMICS, te dicen, óptimamente, no solo tendrías que tener un fluorocromo en cada. O sea, que el PIC 1, o sea, el más intenso, no coincida. Entonces, por una parte está el Similarity Index, que no debería ser mayor de 0,98 y todo, pero luego te dicen, bueno, es verdad, has tenido ciertos problemas, pero porque compartían el PIC, el P1.
Entonces, máximo, dos fluorocromos distintos comparten el pico máximo del detector.
Entonces, si ellos dicen, por una parte está el Similarity Index, pero también te dicen, es mejor que no compartan, no sé tu experiencia si tú cuando haces diseños de paneles intentas evitar que compartan, o porque entonces volveríamos a tener un límite de fluorocromos porque dependería de canales. ¿Tú has combinado con P1 y ningún problema?
Es decir, esta pregunta ya me la hizo Joan, que tienes delante hace, ¿no? Estuvimos hablando ya sobre esto. Estuvimos media hora,
Pero es que es lo que luego ves cosas malas también.
La pregunta tiene mucho sentido, ¿no? Porque al final el detector máximo es el que te va a dar la señal fluorescente final. Los demás lo que te sirven es para que el algoritmo sea capaz de diferenciar las señales. Entonces, claro, hasta ese punto había que ver qué más hay detrás de esto, ¿no? de esta paleta. Yo no me fijo en el pico máximo, ¿no?
Otra cosa importante es ten cuidado con lo que te digan desde las compañías comerciales, porque a lo mejor no son las personas más formadas en este tema y es mejor que le preguntes al experto.
Porque el algoritmo trabaja y ellos no te dicen cómo es. Entonces, si tú haces un unmixing teniendo en cuenta un single color, pero que el single color no está en el, o sea, que ese fluorocromo no lo tienes en el mix, y luego vuelves a hacer el unmixing quitando el single color, es decir, computando ese fluorescencia, te va a cambiar. Entonces,
no preguntes porque eso está patentado, ¿no? Entonces,
exacto. Y luego ya te metes, cada casa comercial tiene su algoritmo. Por eso digo, es que cuando empiezas a rascar, yo cada vez que tiemblo más. Y si nosotros temblamos, ya no quiero ni pensar al pobre predoc que tiene un PI que no le ha enseñado nada o lo que sea y le dice, quiero un panel de 40 colores.
Es que al final trabajar con citometría de espectro completo, he cambiado la nomenclatura gracias a Marco, no deja de tener sus complejidades y al final no deja de ser un algoritmo que se mete debajo de esta curva y te da la proporción de cada uno de los marcajes individuales en función de las curvas que hay ahí debajo. Con lo cual, yo insisto, al final cada tecnología tiene un poco sus pros y sus contras y debe ser muy cuidadoso a la hora de escoger realmente con cuál te quedas. Citometría de masas quizás tienes más flexibilidad a la hora de hacer cambios, pero en cambio esta sensibilidad que tienes en citometría espectral o la velocidad una vez has puesto a punto un panel complejo, pues evidentemente es de mucha ayuda, pero estoy perfectamente de acuerdo que todo el tema de optimización del panel y puesta a punto para paneles complejos, esto necesita tiempo y no se puede hacer improvisando ni saltándose pasos, porque si no vamos al desastre.
Lo de el análisis de los datos, ya lo sabes perfectamente, ya lo hemos comentado muchas veces, las iteraciones que puedes hacer, ¿no? Eso te puede afectar muchísimo. Y eso luego lo ves en la calidad de las publicaciones, que dices, bueno, ¿esto qué es? ¿No?
Al final, como son
Quiero ver
cosas
los plots,
en las que
no
los
se ve.
citometristas convencionales no estamos acostumbrados a tratar y hay mucha matemática subyacente que no dominamos, debemos tener mucho cuidado a la hora de hacer las interpretaciones de estos datos y usar estos programas. Y aquí la importancia de lo que se ha dicho, de tener un bioinformático, pero un bioinformático citométrico.
De hecho, en el último Congreso Europeo de la ESCA, había un bioinformático que decía, bueno, en la región de Forward Site, digo, quito todos los eventos que haya apilado para arriba, por la derecha, y tomo solo esta población. Y entonces ya estás eliminando megacariocitos y eosinófilos para automatización y células grandes que pueden ser patológicas. Y todo esto son artefactos y se quedan tan tranquilos después de la sesión.
Al final, lo que intentamos nosotros hacer es hacer una tradición biológica de lo que vemos en la citometría, masas, como será. Entonces, claro, el componente biológico tiene esta perfección. Los informáticos necesitan esa parte de comprensión y de ayuda. Afortunadamente, tenemos herramientas para elegir. O sea, no es mejor una ni otra, sino que al final es cuestión de buscar la herramienta que más se adecua. Pero sí que estamos en un momento un poco crítico. O sea, la citometría da un salto.
Una para Jordi, que me ha gustado mucho la parte esta de que mirabas los dobletes y de repente, uy, ya no hay los dobletes, no hay dobletes. Con los que no tenemos native o alguno con imagen, ¿tienes alguna recomendación para decir, hombre, pues intenta, claro, es verdad que hay singletes en los dobletes, pero hay dobletes. Entonces, al final también, si cogieras esa región entera, bueno, harías algún tipo de error luego, tema de fluorescencia, etc. Entonces, no sé si tienes alguna recomendación.
Eso es algo que va a depender del tipo celular que estés estudiando, ¿no? Y de la relevancia que pueda tener que en la región de agregados y dobletes tengas células únicas. Va a depender del tipo de experimento que estés haciendo fundamentalmente. Si estás estudiando células minoritarias, puede que estés perdiendo una proporción importante de estas células utilizando ese sistema de discriminación, pero si estás analizando líneas celulares o poblaciones mayoritarias, tampoco vas a hacer un impacto muy grande. Eso sí que claramente te permite rápidamente visualizar, ¿no? Que ahí tienes donde teóricamente solo había dobletes y agregados, pues tienes un montón de célula única.
Entonces, tú has podido gatear con la imagen las 3 poblaciones diferentes y has podido gatearla que sí es single.
Sí, eso te lo permite el análisis morfológico, además del fenotipado convencional. Y entonces no lo puedes hacer.
Aquí la importancia y el interés de ir incorporando esas nuevas tecnologías para resolver estas cosas que normalmente nos quedaban fuera de nuestro ámbito de aplicación.
Porque luego, si tú con el tuyo, con la imagen, te permitiría incorporar dentro de, o sea, no sé si hace foto a todas las células, por tanto, tú podrías pasar por el paso este que has hecho.
Puedes hacer fotos de todas las células.
Y entonces,
Sí.
o sea, incluirías dentro de tu estrategia de gating, pues, estos 2 parámetros que te permitían gatear las singles, las dobles.
Y es extraordinariamente útil en el estudio de células minoritarias y en el caso de que estamos comparando enfermedad medible residual, ¿no? Entonces ahí es fundamental porque estás identificando, dices, estas células que no me creería que es un marcaje muy débil, que sean células que dices, bueno, este será inespecífico, pero ¡pam! Que veas una célula hiperdiploide Ki67 positiva, como las hemos visto, y que te la miras la imagen y dices, pues sí, pues esta célula es muy fea, ¿no? Muy grande, muy rara, y dices, pinta mal, ¿no? Sí, te permite la rápida identificación de las células patológicas. O confirmar que aquel puntito, ¿no? Es realmente una célula, que es muchas veces difícil. ¿Y esto qué es? Pues ahí está.
El One Channel en el full spectrum tiene la misma, o sea, impacta de la misma forma, porque claro, igual si juntas muchas células en un channel o tipos celulares, luego la huella espectral también se modifica. Y no sé si es más recomendable, menos recomendable.
Si utilizas como Dan Channel un fluorocromo tándem, no es lo más adecuado, porque como tú vas a añadir, por ejemplo, APC67, ¿no? Tú añades 4 o 5 anticuerpos APC67, es posible que entre ellos haya diferencias en la huella espectral. Es más fácil que haya diferencias en estos que no en un APC o en un P o un FITC. ¿Vale? Entonces, nosotros hicimos un Dan Channel en uno de los proyectos y sí que es verdad que nos dio algunos problemas y quitamos uno de los 4 que tenía y los pusimos en otro. Es, como todo, cuestión de probarlo. O sea, dependiendo de los marcadores que ponga y de los fluorocromos te puede funcionar.
Pero si quieres descartar un montón de, si estás hablando de tejido, quieres descartar un montón de tipos celulares muy diversos, esto es igual una locura.
No, pero, claro, si pones muchos anticuerpos y lo metes en un Dan Channel, puedes tener un problema. A nivel de huellas, seguramente si pones muchos, sí. Pues, nosotros los que hemos hecho ha sido con pocos, ¿no? Y
además el Dan Channel nace de la poca disponibilidad de detectores, o sea, tienes mucha más ventaja usando un equipo con muchos detectores porque te ahorras el problema asociado al Dan Channel.
Hay que tener mucho cuidado porque a veces te encuentras marcadores, por lo menos en sangre periférica, en medio de la ósea, que tú no te piensas que van a ser positivos y te das sorpresa y te los quitas con el Dan Channel y estás perdiendo células. ¿Sabes? Pero bueno, al final siempre hay que saber la biología antes de...
Y aparte que cuando tienes células diferentes, con niveles de autofluorescencia diferentes, aunque cada vez los softwares de citometría de espectro completo tienen mayor capacidad para tratar con autofluorescencias múltiples, pues no deja de ser complejo y no deja de ser un problema porque no todas las células tienen el mismo background. Por esto aquí la gracia de usar estas otras alternativas, ya sea por imagen, ya sea por sistemas de proteometría de masas que no tengan
Más simple, hay un artículo muy antiguo en CYTOMICS que añades azul de tripán para eliminar autofluorescencia. Pero tú no lo puedes utilizar con lo de masas. Simplemente añadiendo azul de tripán para eliminar la autofluorescencia, así de simple.
Sí,
Esto me dio en el año 1970 y pico.
Que no te haga quenching de otros marcadores fuego también.
No, pero sigo un momento más. Imagínate que te interesan las células T o linfocitos, ¿no? de un tejido. Mi pregunta es mejor también identificar tipos celulares que van a ser muy abundantes en tu tejido, que no quieres, pero los pones, pues igual no en un dump channel, sino en 2 o 3 dump channels, para, digamos, limpiar y quitarte, sobre todo en tejido, no sangre, que es lo que estoy. O no, céntrate en las tuyas y pasa del resto.
Cuanto más limpies, mejor, eso siempre. Siempre. Porque,
O sea, si puedes identificar epiteliales, fibroblastos, no sé qué.
claro, porque además, incluso puedes tener uniones inespecíficas, depende de los tipos celulares que tengas acompañantes y que no te interesen. Igual, al final, si tienes que llegar a decir, en pulmón, riñón, por ejemplo, pues claro, ¿qué ocurre? Pues tienes unas autofluorescencias impresionantes, pero no tienes una. Tienes 4 o 5 distintas. Claro, tienes que buscar el equilibrio, o sea, tienes que analizar tu muestra. Lo primero que hay que hacer es pasar tu muestra, diseñar y ver qué patrón de autofluorescencia tienes. Y a partir de ahí, empezar a programar el panel.
Nosotros, por ejemplo, siempre ponemos un marcador de DNA, que puede ser una sonda vital, y eso te quita un montón de ruido ya.
Pero como
Te ayuda a discriminar muy bien las células de interés, incluso para células muy minoritarias, ¿no? Imagínate, estás estudiando un líquido cefalorraquídeo y sencillamente añadiendo el marcador de DNA identifica rapidísimamente las células. Luego, si las puedes visualizar con otros resultados que tengo, se ven clarísimamente que esos son células y esos 4 puntitos no es nada inespecífico, ¿no? Son células verdaderas.
Y la otra opción es, si trabajas en tejido, es ir directamente a un sistema de imagen, como el Hyperion que comentaba, donde tú no tienes autofluorescencias, donde tienes muchos canales abiertos para poner muchos marcadores diferentes, identificar con propiedad cada una de tus células sin perder la información de morfología.
Yo no vendo páginas.
Haber puesto una cosa cada uno de color de
Vamos a continuar con la sesión. Vamos a empezar la sesión desconcertada. La primera presentación correrá a cargo de Gloria Bartuz, Amateur, Standard Biotools, cuando quieras.
Vale, gracias.
Wonderful. Good. So I was told to do the talk in English. My name is Gloria. I'm based in Barcelona. I am the FIS here in Iberia, so field application scientist for Standard Biotools, former Fluidine, and I'm actually the team leader of the FIS in Europe for the company. A little bit about me. I did my PhD in IrsiCaixa. I was trained by Marco on flow cytometry. I fought with FITSI, PE on an LS or two. I went to my postdoc in Germany. I fought with 15 color panel markers on Fortezas, Arias. So I got my kind of my share of fighting with cytometry. And I decided to go to what you guys think is a dark side. I also thought it was a dark side. It's getting a bit brighter now. And I went to a company as a field application scientist because I realized in my postdoc that what I like is to teach. I really like to teach people. I really like to enlighten people. And part of what we do is actually teach people how to use our technology. And this is something I really like. So that's a little bit about me. One thing that I wanted to ask you is who likes to build Legos? Please raise your hands because I'm one of them. Great. So we have a Lego set that is our system. So it's XT and Hyperion here. So the only thing you have to do is scan this code. This code will be also at the end of the talk. So if you miss it now, it's going to be at the end of the talk. It's a competition also. So the one that has the higher the higher points here today is going to get the Lego set. Okay? So raising your competition levels here. And yeah, I'm going to leave it for 30 seconds for you to just scan the code. Please don't play during my talk. I mean, I would appreciate if you listen to me too. But yeah, that's for you. Good. It's also at the end of the talk if you guys want to use it later for the Q&A section. Good. Then I see some points still raised, so I'm gonna wait for you guys, and then we're good to go. Wonderful. So I think this was already previously introduced by Jama nicely. What you see is what you get. This is where you see a cytometrist on the flow, even as we said, like on a full spectral. We have fluorescent labels, wonderful labels, but unfortunately they have chemical and physical properties, and the spectrum is not always very tight. While with EMC metal labels, we just see sharp signals, do not overlap, easy to build panels, right? This is us, this is Standard BioTools. So we nicely incorporated now recently, I'm not sure if some of you know about it, SomaLogic. It's a technology that allows you to actually detect it's proteomics in sample urine, in samples like urine, like blood, like cerebral spinal fluid. So it's kind of going into the ultra high flex serum. They can measure up to 7,000 to 11,000 proteins at the same time. So if you're interested, just stop by the booth. And then we have also the genomics part. We also have like high throughput nanoscale NGS with our microchips. But what I will focus on today is on the part of flow cytometry, just a little bit, because Jama gave a really good introduction about it, and the imaging part, that it's actually really beautiful with beautiful images. So these are our two phases. We have the flow phase, and as the cytometry also, you'll see that there's no negative signals, 0, 0. We don't have compensation, which means that we don't have to go to 10 to the -3. We don't have this spreading signals. 0 is real, and what we define as a nice signal is over 10. This is our nice signals, right? So this is what we see as in flow. And then we have the imaging part. For flow, we see more than 60 markers. So this is the little update compared to what Jama said earlier. Recently, we launched new metals. So now we can go up to 60 markers. If you need them, you don't always need them. And then there's the imaging part where we can go up to 40 plus markers, okay? So the agenda for today, as an application scientist, I like to show data. I like to show papers. They speak for themselves. So the first ones will be some application examples. Then I'm going to move to just basic background on the XT and then the imaging mass cytometry. The first one is just to show you the evolution of how many papers were published using our technique. So as I say, it's been like increasing since 2007 when the first CYTOMICS was released. And then there's the new generation systems that were released later on. And statistically last year, 96% of the publications that have more than 20 plus markers were using our technology. And EMC, so imaging mass cytometry, is the most used one for more than 20 plus markers. But this is papers published last year, right? So from last year. And we are present in 232 clinical trials. So when your technology is used in clinical trials, that shows how stable the technology is. And papers are in quite some different fields. So it was quite difficult for me to choose some papers that would be interesting for you. I hope I did well. The first one, for the ones that do not know, I did my postdoc in NK cells. I have a huge love for NK cells. They're very dear to my heart. And this is a paper that is a beautiful one, published in 2020, where they used memory-like NK cells that were, induced with cytokines to try in a clinical phase 1 trial with patients that were that had leukemia, and they were in last stage. They had no other hope. That was the end. So they had no other therapies. They failed everything. And they tried this memory-like NK cell phenotype infusion. So what they did, they took blood from an HLA, obviously, haploidentical, otherwise NK cells will be crazy about it. They took the NK cells, they activated them to become memory-like NK cells, and then they infused them to the patients. Why they used CYTOF? Because they wanted to analyze everything on the samples. So they went with 70 parameters, they divide them in 3 panels, and they divided in 3 panels. They had like a backbone that was common to all the panels, and they added extra markers to each of them. So when they did the analysis, they could cross-react all the different panels. So at the end of the day, they had 70 parameters altogether. So what they obviously show is that the donor NK cells, the donor plus activation, and the memory-like NK cells from the donor at day 7, they actually cluster very differently. So that means that there's some evolution. The result is that there's a memory-like NK cell signature that it's very specific. And as you see here, there's treatment failure responder. So the responder did not have on their NK cells, CD8 positive cells, NKG2A positive cells did not exist. While the treatment failure, the ones that failed the treatment, had an early population that showed quite impressively there, showing both markers. So they knew that these patients will fail this treatment. What is important, actually, this is something that really helps us when you are in a company. You need to, you find some things that will really give you hope, is that this patient that was responder became eligible for hematopoietic cell transplantation and actually survived. So that was a patient that was actually on the verge of dying and was able with this memory-like NK cell therapy to go into this next phase. After that, they built a phase 2 clinical trial to try to understand why that happened and trying to reach more patients. Another one was CD4 positive CAR T cell infusion. So there were two patients that were cured, and they were in 10-year long remission. No one really understood why. And they wanted to map the complete fate. So what happens there? This is pure discovery. They didn't really know what they wanted to look for. So why they use CYTOF is because they wanted to identify CAR T cells. So they wanted to go into the T cell population, and they wanted to go very deep into it, not only in the phenotype, but also in the functional status and even transcription factors. So the results in cover, so this is a UMAP, right? And this is another one. Basically, they show the same thing. This is the 2 patients that are clustered together, and this is time. So the longer you go here, this is the time progression, right? It goes from, actually, let me recheck that 1 second. It goes from here to here. So basically, they rediscovered that there was the CD4 negative, CD8 negative, Helios high, gamma delta CAR T population in earlier time points that was specific. CD8 positive CAR T cells diminish over time. And as you see here, CD8 positive CAR T cells are actually these ones that are over here, like the reddish, these ones actually disappear over time. This is years. And the one that is predominant is the CD4 positive, Ki67 positive CAR T cells. And the fact that these two are predominant in both patients actually indicates that if you have this signature, you're going to be in complete remission. Apparently, they're still undergoing. Next one, SARS-CoV-2. This is a paper coming out of Andrea Cosarizza's lab in Modena. They're working from Sara Biasi. They're working on trying to understand the vaccine response in MS patients that are treated with different disease-modifying therapies. So it's kind of a complex situation because you have all these other immune treatments on top of the immunization. Why they use CYTOF is because they wanted to build like a very big panel, and they wanted to go functional and metabolic. So they have a lot of antibodies that are targeting phosphoproteins, which are sometimes not easy to undercover with other techniques.
So the conclusions, they went for antigen B cells, antigen T cells, that there was detectable functional immune response, and that 2 drugs generate a unique immune response. So that was the conclusion of the whole paper, that it's actually quite big. And they identified a SARS-CoV-2 immunological signature. And the final one, sorry, this one is, this is wrong actually, this paper is about this is more, now I'm switching to imaging. It's not only about understanding what is in the blood, but also what happens in the tissues, right? So this one is about understanding the tumor microenvironment structure in patients that actually needed to understand what happened in that part, or not only in function, like in surface, but also apoptosis, proliferation, and intracellular. So they discovered that there were 10 recurrent DNA structures that varied quite a lot, and this is in breast cancer, and that they show somatic alterations that will go into different subtypes of breast cancer. That is interesting because that could predict clinical outcome. Going back to that, I need to show you a little bit what we did too. We recently developed a new panel that is the immuno-oncology panel that spans for tissue architecture, basic immune, stromal, myeloid, lymphoid, cell state. So this is only 31 markers, and I say only 31. This is in imaging, and you can add up to 9 to 10 extra markers if you want to, if they're not enough. And these are all the list of the different markers that are on this imaging mass cytometry panel. So it's composed of different panels that when we mix them all together, they work nicely. Okay? Good. Another one that is interesting also is how our brain reacted to COVID inflammation, COVID-19 infection. And the idea was to try to understand, unfortunately, at brain autopsy, because getting brain samples is difficult otherwise. They did like a 38 marker panel. Still, they went on the edge. They wanted to understand what CD8 T cells were doing in the brain and what happened to the myeloid cells. The results, and this is just very long story short, they built a brain immune map to understand the activity in microglia, what happened there. And then they saw that actually, very similar to other diseases in the brain, it was more on the expression of PD-1 on CD8 cells that were interacting with the myeloid, the microglia clusters that were expressing PD-L1. And that was generating these huge nodules on the microglia, and obviously inflammation, and obviously we know the end for some patients. Linked to that, there was a development of a new panel that is a human panel, and there's a mouse panel, and they both overlap for some markers. So if you're interested, you have a preclinical study, you want to go into clinical study, phase 1, so you have something that you can actually overlap. And linked to that, because when we are doing, and this is something I will explain you later, when we are doing imaging, we need to extract the data, right? It's not about pretty images, it's about understanding the single cell data. So that's why we have one kit that is called EMC segmentation kit. So this kit is 3 antibodies that stain universally mouse, human tissues, and you use them for later data analysis and segmentation of your cells, your single cells, okay? Is that all clear? Any doubts so far? Okay, good. Next step, at the site. Going back to that. So this is what I showed you earlier. It's the system that Jama showed you is the Helios, which is the 3rd generation system. This is the CyTOMICS, that's the 4th generation system. There's no difference on the data you get out of it. So you still get an FCS file that it's clean. You don't have to do anything about it. This FCS file, you can use it directly to do your analysis. There's no deconvolution, no compensation, nothing you have to do to your samples. The only difference between this one and the former generation is that this is automated. So you can run your samples automatically. You don't have to, there's a carousel, obviously, like this is something that is not new in cytometry, but there's a carousel. You can control the system remotely, you can connect to it. It can send you emails or whatever warnings if something is off with the system. So, and it can shut down automatically. So you don't have to be in front of it to use your samples. And going back to what we can offer. So this is a cell, as we know. So there's membrane markers that we want to stain. There's cytokines that we also want to stain. There's phosphoproteins, because this is for the metabolic states. Phosphoproteins are very, very interesting for a lot of different diseases. They are extremely important. There's transcription factors. These will help you to identify some cell types, actually. So without them, you cannot really work. And right now, we can even detect mRNA. So this is everything you want to detect. And this is what you can detect with our technology without any problems. Why? Because the metals do not care about fixation reagents. They don't care about light. They don't care about being frozen. So that's why we can freeze our samples. That's it, once they're fixed. They don't care about permeabilization reagents. So that's it. That's why it's so big. We have in Catalog more than 800 plus antibodies for flow, more than 300 plus antibodies for EMC. But if we do not have any antibody, your antibody of interest, your clone of interest, then you buy the antibody purified and there's a labeling kit. You can do it yourself at home. Well, at home not, in the lab, hopefully. And it takes like 4 hours, but because there's a lot of incubation times and it's very easy, straightforward, very efficient. So it's very flexible. You can build your own panel. Normally, I would say, please contact me so I can help you to build your first panel. I can teach you how you build a panel, and then we just do it together. In my 4 years being an FIS and now being the team leader of the FSs in Europe, I have visibility over all the panels we build. We never had to change a single antibody in any panel that we built, meaning that what we do in silico transfers to what we do in vivo. Okay, so what you build first, you don't have to cross-validate anything. You just do titrations. Titrations are very important because otherwise, without that, you may be overspending money. But not only that, is because maybe your signal is overshooting. But that's it. And titrations can be done in bulk. So we calculated that with 12 tubes, we can titrate your 40 marker panel. Only 12 tubes. That's it. So between building a panel and having it set up, a month, if you have the antibodies. Okay? Good. But titrations are important. That's the one thing that I cannot emphasize anymore enough. Good. And now I'm going to focus into the imaging part. So we all know that the context is key. It's not about knowing which cell types we have, but also where they are located, right? So we know that what happens in a tumor, it's super heterogeneous, but not only in tumors, in any kind of different tissues, right? You go to the liver, you go to the kidney, you go to whatever, you will have a huge variability of cells that are, depending on the context, they are very, very different. So the interactions and the spatial relationship between them is extremely important to understand. That's why I like the Legos. So this is what we can get with bulk analysis. But obviously you need to try things and you need to classify. We're humans, we love to classify stuff. We love to put things in boxes. So we classify by colors and we get single cell analysis, which is cytometry, right? And single cell RNA-seq. But the next thing is, what do I do with that? How do I know how they interact with each other? And actually at the end, this was a house. So that's what spatial techniques will give you. This is the Hyperion XTI. So it's a system that connects to the cytometer. We only use the cytometer now as a detector, right? There's only one single detector, it's a mass spectrometer. There's only one single detector. So we use the cytometer as a detector. And here in this small window, we introduce our tissue slide like that. And it's going to automatically acquire the data and prepare your sample and get you the data out. So no interfluorescence, 40 plus markers. You can use RNA scope with the technique. We have the cell segmentation that I already told you. You can do batch slide staining and storage. So similar to flow, in flow, we freeze the samples that are stained, ship them to a cold facility. Well, we freeze them at -80. You can keep them 3 months in your freezer. So you don't have to rush, you ship them to the cold facility. For EMC, actually, the storage, it's actually years. So you stain your sample with your tissue. You cut your sample, you stain fresh, and then you can store it for 1-2 years at room temperature. And then you have time to send it to a co-facility and do your acquisitions. So you don't have to own the system yourself to do it. I mean, for us, it will be better if you own the system because that means that we earn money. Paolo will be very happy about it, but you know how to own the system to actually run it. And finally, if you have this one, that means that you have this one. So you can do what a lot of co-facilities and labs are doing, which is dual mode. Depending on the day, I need to do imaging. Depending on the day, I need to do cytometry. It's 10-15 minutes to switch modes. So it's quite flexible in that sense. How does the protocol look like? You get your antibodies that are labeled to metals, again, out of our catalog or because you label them yourself in the lab. You stain your tissue with the antibodies, all 40 antibodies, 41 antibodies, all at the same time. And then you measure them using the XDI. You collect the data, and we perform the data analysis. The idea is that, as I said, you can batch stain, you can ship or store, and then you acquire. So you don't have to rush to the instrument. How does the workflow works for both systems? And I like to put both together so you see the comparison. So we can do the panel design in under 2 days. We can swap the markers with very minimal overlap, any of the both technologies. We can do simultaneously staining with RNA co-detection. So that will be more for imaging, still not up to speed for flow, but it's just a matter of someone trying it, to be honest. And we can stain samples are stable for months or actually, well, months in -80 or years if it's a tissue slide. You can do co-simultaneous analysis of 40 plus or 60 plus markers. 40 plus will be for imaging, 60 plus will be for cytometry, any tissue type, and then you can make quick decisions. So at the end of the day, data analysis, I agree with what it was said before. I feel like we do not have the knowledge. Not all of us have the knowledge to do data analysis. Even myself, I'm getting trained, like every month we have this kind of trainings, and I'm still not up to speed. For some of the things you don't need a bioinformatician, but it's nice to have someone that has the knowledge to do all this computational analysis together. For imaging, I will show you later, we are developing a software in-house that will help to do this data analysis workflow. Right now, what we are getting out in the system, like in 1 month, it's a slide loader, because if we are going faster, if we are going like, we are so efficient, we need to get like some kind of automated slide loader where, so no, we don't have to have a master student, PhD student, because that's what it happens in labs, going there, changing the slide and then going out, because that's what happens. And then we felt really bad about it because we all were there. And yeah, that's a slide loader. So you can automatically program that. You can control the system from home too. It's very, very ideal. And obviously if you have a barcoding, it has a barcode reader. So everything is very automatic. So as I said, it's not about pretty images. We all like pretty images, but it's about analyzing what happens in the images, right? So the idea is to extract single cell data out of your image to build t-SNE. We use t-SNE because there's not so many cells, right? It's not like you have millions of cells. In these RIs, you have some few cells, but you'll see we are getting bigger and bigger. So now we might need to adjust these algorithms. You cluster that, you gate on the cluster interest, and then obviously you can visualize back. So this is what we call the round trip analysis. You don't only visualize, but you also are able to calculate the distance between 2 cell types, 3 cell types. This is the idea, right? You need to understand if B cells are close to T cells, or if PD-1 positive cells are interacting with PD-L1 positive cells. That is what you need to understand. So that's why these neighborhood analyses are important. How do we do that? There's a free of charge workflow that allows you to do that. The first one is view, validate, all the stainings work. So something that failed, we need to go back, understand why it failed. Maybe the antibody titration was not the perfect one, whatever. So this is done with our system, MCD Viewer, free of charge. And then we have the segmentation. There's quite some algorithms that are now developed for that. And they're, again, free of charge. And then there's high order analysis, like what I show you, t-SNE, phenographs, clustering, everything. This can be done with IstoCAD, also free of charge. If you have a bioinformatician, he will do everything, or she will do everything in R. So you don't even need to worry about that. But there's also some commercial houses like Indica Labs or Visiopharm that develop their own solutions too, that work both with fluorescence and with mass. Last thing I want to show you is our new imaging modes that we released a month ago. It's going to be official in July. So imagine you have a brain tissue. And again, all of those are co-staining, just that our human eye cannot see the 40 markers together at once, right? We will mix all the colors together. We will not see anything. So it's a panel with 40 markers. And then we just decided these combinations for you to see. So we have a new mode that is called tissue mode. This allows us to do the whole tissue slide. We do that at 5 micron resolution. And it helps to decide on structures on your tissue. So where do I have my immune cells? Where do I have like a tissue, like a lymphoid structure like that? Where do I have a cancer structure, like, well, kind of like a positive structure? And for that, there's pixel-based analysis. 5 microns do not allow you to do single cell analysis anymore, but it's pixel-based analysis. And this is where we are developing our in-house solution to help you analyze that automatically, very similar to the MDPA. Next one is what we call the preview mode. So preview mode is a solution for us to just preview the sample. So we just go in 1 micron out of 25, just to understand more or less how the sample looks like. And then it supports the scientists to decide which ROI do I want to acquire? Where do I want to focus? Is it important for me to focus here, or do I want to focus here? And then we go into the cell mode, which is the single cell one. And this is our region of interest. Historically, this is where we were focusing. We focus at 1 micron resolution. Now with the XDI, we can go at 0.5 micron resolution. So it's subcellular, and we can actually see some quite structures. And right now the ROIs, this region of interest, are increased up to 10 by 10. It used to be 2 by 2. This is the big jump that we did. We increased it by 5. So this is just for you to get an impression. This will be preview mode to understand what happens in the tissue. Where do I want to focus? And this will be the cell mode, where you see the individual cells, and then you can see the different, interactions. So just for you to understand, a quick summary. This is the XCI. I mean, it says here 5 times faster because you don't know the previous system, but the previous system was 5 times lower. So this is 5 times faster. And much limits. So we are actually better now at detecting dim markers. Again, PD1 is one of them that it's very difficult to detect in imaging. So this one is there. Walk away acquisition for flow cytometry mode and also for imaging mode. And there's the new 3 modes of acquisition. Review tissue. Tissue is the one that we actually are focusing on now with the 5 micron resolution, going through the whole structures, and the cell mode that it's the 1 micron resolution going into regions of interest. So experimental design. Minimal predictable spillover, no impact of light, fixing permeabilization reagents. Stained samples can be frozen or kept at room temperature, shipped to a cool facility. You have one that it's in JAMA's for cytometry. JAMA is there. If you need information, just come by the booth because we do have some list of places where you can send your imaging ones. Flexible, fast panel design, and we can do this point. Automated acquisition, familiar workflows. So the staining protocols are very similar. No cyclic staining for imaging, and we can do more than 100 samples per week in imaging. And data analysis, no background, no fluorescence, especially very important when you're doing monocytes, when you're doing tissues, brain, liver. I fought with liver quite a lot. So liver is super autofluorescence. They really like to shine the livers. So if you have some nasty tissues, it's imaging, mass cytometry is really helpful. And the protocols and the formats, the pipelines are very, very similar. No compensation, no deconvolution needed. And you get a direct FCS file out of the system, or EMD, this is for imaging. And we, as I said, we are developing solutions for pixel-based analysis to help automate all this data analysis. Going back to data analysis, this is something that we know as companies, we know we need to focus there. And that's why we are putting a lot of efforts from our bioinformaticians. And how can we help you? Hopefully, we can help you. There's a lot of protocols that we have online. We can do workshops. We can, as I said, I'm an FIS because I like to teach, because I like to explain. So happy to do anything. So just feel free to contact. We have this free panel design service. We are also there to troubleshoot. If something happens, we are there to troubleshoot. And if you need more tips on troubleshooting, just contact. And that's our emails. So as I contact your FIS sales, I'm your FIS in Spain. So that's my name, that's my email. Then that's Paolo Santino, my colleague that is in the back. He is my regional sales manager in Iberia. So feel free to contact him. We can discuss your project goals. Pre-work, and I agree a lot with, well, I agree with everything they said before. Pre-work is important. Understanding which markers you want to put, where you want to put it, why you want to put it, which clones you want to use. And if you already have some knowledge about the clones, it's important. So discussing projects before doing panels and understanding that you have all the samples, not about, I mean, if you're doing a disease, it's also about understanding if you have the right controls on the healthy controls or something to compare to, right? So we are scientists, so we actually can help you with project discussions. Designing your panels, obviously, this is something where we are experts and we can teach you. We have an online system. It's very easy to learn. In less than 2 hours, I can show you how to design your panel. So up to you, and order your reagents. And I leave here this light up. So if you still want to play, this is the logo. And yeah, thank you for your attention. Do you have any questions, burning questions? Yes, John.
I have a question that I didn't get. So you with your slide and analyze the image.
Yeah.
Then that very same slide of tissue sample is pulverized into the.
Yeah, so I didn't want to go in detail because it was a bit too technical. The way it works is that there's a laser inside that tower. There's a laser. And what this laser will do, imagine that you have here, this is your slide. What the laser will do is will ablate line by line your tissue. And this one, in that case, if it's 1 micron pixel resolution, this 1 micron will be sent to the system. And because we know the time between and the coordinates of that 1 micron pixel until it hits the detector at the end of the XT, this is the live reconstruction of the image.
So you can correlate the image.
Yes. Yes, actually it's done live. So you don't even have to do anything. This is, as you see the ablations happening, you actually see the image being reconstructed at the same time. It's beautiful to see. So in the previous system, it was something that you could see it was going slower. And now at 800 Hertz, it just goes at 800 Hertz. So it's quite fast. And then you see the reconstruction live. Yeah. But obviously you cannot see all the 40 markers together. So what we always monitor is DNA and some structural markers to understand that everything is properly working.
Okay.
Yeah, wonderful. Thank you.
Move to the next presentation that will be about by Maria Carboni on MapTech. Your turn.
So, hello everyone. My name is Maria Carboni, and I'm presenting MapTech today. I'm a field application specialist at MapTech. Today we will talk about Fluorospot and the applications that it has in immune cell research. How you can use it. Thank you. How you can use it in your immune cell research. So let's talk a little bit about MapTech and our story. So we are a company founded in Stockholm in Sweden around the mid-80s, as you can see. And we were founded by 3 experts in making monoclonal antibodies and specifically matched pairs. So they developed those and they used them to make different kits such as ELISpot and Fluorospot kits, also ELISAs. And then also as of 2018, they started developing, I'm not sure how it uses. Yeah, as of 2018, we started launching our own readers for ELISpot and Fluorospot analysis. The latest reader is here, 2023. We have the 2nd generation of readers, Astor 2 and Iris 2. And we are very happy to be distributed by lab clinics in Spain and Portugal. So this is where you can find our products, kits and readers. These are our core assays. We have ELISpot and Fluorospot, where you can measure analyte secretion, such as cytokine secretion or antibodies in a single cell level. And we also provide ELISAs, where you can measure your analyte concentration in a solution. And as of recently, we also have FosciSpot, which is a way to detect virus-infected cells. But today I will only focus on Fluorospot and give you different options how you can apply this assay in your research. Starting off with what Fluorospot is basically a multiplex version of ELISpot. We start here at step 1 by coating a 96 well plate that has a PVDF membrane on the bottom of it with antibodies. So PVDF membranes are very good into absorbing proteins. They are high protein binding. So you can activate that with ethanol, for instance, and then you apply your antibody of interest, and this coats the surface. After the coating, you can add your cells and the stimulation. So you add them together and you incubate them for an amount that is appropriate for the kinetics of the analyte that you are detecting. For instance, for interferon gamma, we allowed between 18 to 24 hours versus a granzyme B or perforin that needs longer incubation time.
After this incubation time, you wash away the cells are not present anymore, and you just have basically the coating antibody here, the capture antibody that has captured your analyte that was secreted by your cells, and then you add your detection antibodies. So this is what happens here. We have the first step, which is an anti-analyte antibody, it would be interferon gamma, for instance. This will bind the analyte, and then we can detect this antibody using a sortavidin conjugate with fluorophore. So this binds to the biotin, or we could have a tagged antibody that could bind to specifically to the specific tag, and then has a different color, fluorophore, so you can detect the different analytes in your fluorospot well. The last step is, of course, the analysis here, and we have a reader that can do that very easily for you. If we look a little bit closer on what you get after analyzing a fluorospot plate, first of all, you get the total spot counts per cytokine. So it's a little bit like having 3 different ELISpot all at once. So you could have interferon gamma, IL-20 and F in this particular example, and you could make different graphs using these data that are generated. Another way where you can use a fluorospot and the analysis that you get is by measuring the co-secretion of analyte. So for instance, if you look closer here in the image, you can find some spots that are produced by only one analyte, some that are produced by 2, and some from 3. So you can plot that again and measure the polyfunctional T cells in your cell suspension. For doing that, you would need to have a very good algorithm that should detect very accurately the spot center, as well as the coordinates of the spot into the well. Once you have that, you can tell that a specific spot, that the spots align and that this came from the same cell, or it could be that the spots do not align. You have one red here and one green here that do not align, so these do not came from the same cell. This is very important to do, especially when you're assessing polyfunctionality of T cells. You want to know exactly if the 2 analytes came from the same cell or not, because it could be that they are so close together that it could be mistaken as one. But a good algorithm should be able to differentiate them and tell you if you have a polyfunctional cell or not. This is what we wanted to do, and this is what we collaborated with mathematicians in KTH for. KTH is the Royal Institute of Technology in Stockholm. And they created a very complicated mathematical model predicting the dilution of an analyte into solution, also by using Bryonian movement principles. So they created this algorithm for us so that we do not base our analysis just on image processing, but also on signal processing. So with this algorithm, we can now see the whole peaks of the signal. You can imagine that the analyte is piling up as sand, and you can measure the whole peak of this. You can see, for instance, this single spot here came from 1 or from 2, and in this occasion, it did come from 2 different cells. So this is the most accurate algorithm that exists in the market out there, and we do not keep it a secret. This is all public, and there are publications on that from KTH, and you can scan this code if you want to read more about it. But because of the algorithm and the possibilities that it gives, we are also now able to detect the relative spot volume, which I will talk a little bit more later. It's basically the volume of the whole peak. First of all, these are our readers. We have the new generation, as I mentioned, Astor2 and Iris2. Astor2 is an ELISpot only reader, whereas Iris2 is an ELISpot, fluorospot, and foci spot reader. So you can do all 3 with this. And they both use the same software and the same algorithm. So now we're going back a little bit on the relative spot volume, or RSV. And this is the 3D volume of each spot. You can use this, it doesn't tell you the picograms of the analyte, but you can use it to compare different spots that you see. For instance, here, you can clearly see that this is a much bigger spot comparison to the one on the left. And with RSV values, you can tell that this is actually 10 times more intense and bigger in volume comparison to the smaller peak here. This has a lot of applications, the RSV values. For instance, if you want to compare different stimulation conditions or patient cohorts between them, or you can compare different cell types and immune cell subsets. This is RSV in action. If you would to just compare the spot forming units in these particular wells, you would see that the spots are very similar in the count. However, if you wanted to still have a conclusion on which peptide was able to stimulate your T cell more, then you would have to use the RSV values to plot them, and then you would see that actually the top peptide 1 had brighter spots, more intense spots, which means that the peptide 1 stimulated the T cells to secrete more amount of the analyte of interest here. So next, I will go more into applications, specifically for T and B cell research. Starting off with T cells, when you use fluorospot in T cell assays, you can assess the polyfunctionality of T cells and the different cytokine secretion levels with RSV. Polyfunctionality is something very important to always consider in a study, just because there are many different reviews that have been published about how polyfunctional T cells are able to control an infection better than monofunctional cells. And polyfunctional T cells in this fluorospot is basically the T cells that have secreted more than one cytokine. You can see that this plot, for instance, has used MFI for flow cytometry. This is traditionally what people were using to assess the polyfunctionality. But now, since we have introduced this RSV value, we are able to assess polyfunctionality of T cells also with fluorospot. This is a case study from a SARS-CoV-2 research where they used RSV values and they assessed polyfunctional T cells. So in this example, we have a cohort of individuals that were recovering from SARS-CoV-2. They are 5 months post-symptom onset or 9 months post-symptom onset. And this cohort was assessed to see if the T cell immunity was persisting after the infection, and also to see if the quality of memory T cell response was good or bad. So they set up a fluorospot assay to assess these questions. And as you can see, they stimulated their cells for 24 hours with SARS-CoV-2 peptides. And after that, they washed away the cells and they detected the capture analytes here by using the detecting method that I mentioned before. The readout that they could have is the number of responding cells, the relative spot volume, and also the co-secretory profiles. Going into the results, we can see that they plotted, this is a plot for each patient that they did, seeing the monofunctional and polyfunctional cells of this patient. This is after 5 months post-symptom onset or 9 months. And they used then the one that are highlighted with pink here to make a new plot to just assess the polyfunctional T cells of this patient. So they were able to tell that, for instance, the COVID-19 patients had many more polyfunctional T cells in comparison to the healthy control individuals. Now, since they had the RSV values on hand, they were able to dig deeper into their data and see how much of each analyte was secreted. So they made this plot where, for instance, if we look at interferon gamma secretion, they could able to plot the interferon gamma that was secreted by the polyfunctional cells that secreted all 3 analytes, so IL-2 and interferon gamma, TNF and interferon gamma, or only interferon gamma. And they were seeing that there was a common trend among all the analytes, that the polyfunctional cells, the more polyfunctional they were, the more analyte they were secreting. So they found that it's not just that they secrete more analytes, but secrete a larger amount of each of these analytes. In total, they found that there were polyfunctional T cells found in all of these patients using the fluorospot assay, and they were able to tell, again, using the RSV values, that the amount of cytokine was larger as well. Other potential applications would be to test T-cell cross-reactivity between different SARS-CoV-2 variants, for instance, or they could compare T-cell functionality and cytokine secretion between different doses of vaccines or between different infected individuals. I have another case study to show you. This is on cancer immunotherapy. So this is a study where they wanted to compare 3 different CAR constructs against ovarian cancer. This is specifically directed against mesothelin, a very good target for ovarian cancer. So here they had a mouse model. They inoculated the mice with ovarian cancer, specifically from a cancer cell line SCOV3. And then infused them with CAR T cells, human T cells. And they were harboring 3 different CAR constructs, that they were different for the intracellular domains that they had. So they wanted to compare these 3. They harvested the spleens and they isolated with cell sorting the CAR T cells from them. And they found after all this elaborate experiment that they didn't have enough cells. So they had to do something in order to get the conclusions and the readout they wanted with a limited amount of sample they had. They did 2 methods to assess the functionality. First was a flow cytometry assessment for the exhaustion markers, and the second was a 3-color fluorospot. And they actually did that by co-culturing these CAR T cells with the SCOV3 cells again, so rechallenging these CAR T cells directly in the fluorospot plate. And they were analyzing for interferon gamma, granzyme B, and TNF alpha secretion. They were able to use only 800 CAR T cells per well, which were co-cultured 1-to-1 ratio with SCOV3 cells. And these are the results that they got. Here they have compared the 3 different CAR constructs, and they saw that the M1XX CAR was the one able to secrete many more cytokines and was much more potent in comparison to the other 2. So they were able to see that that about half of these CAR T cells were actually able to secrete all the 3 cytokines, and they were also managed to see that they had many more polyfunctional CAR T cells with a specific CAR. So at the end, they were able to conclude that the M1TXX CAR T cells were the superior effector cell, and the specific CAR was the one to move forward for the clinical trials. They saw different advantages for using a fluorospot. They were able to use the low number of CAR T cells that they had. As I said, 800 cells per well only. They were able to also do a longer incubation time to detect granzyme B. This is an analyte that cannot be easily analyzed with flow cytometry, for instance. Here, flow cytometry uses CD107A, which is a surrogate marker. But with flow cytometry, if you incubate for a long time, you can see granzyme B. And you are also able to detect the superproducing CAR T cells with a relative spot volume. Other potential applications of that is, again, to compare different CAR constructs or to do different potency assays and assessments to see if your TILs or CAR NK products or other cell therapy products are potent enough. Now, going into the B cells. With B cells, you can detect antigen-specific B cells or dig again a little bit deeper to find their isotype and subclass. So this is a little bit different depending on, it's a bit more complicated than the T cell fluorospot because you need to understand the difference between the plasmablasts that are already secreting antibodies and the memory B cells that need to be stimulated in order to secrete antibodies. So if you are interested in the memory B cells, then you need to pre-stimulate them with what we call a stim pack, which is recombinant IL-2 and R848, toll-like receptor 7/8 agonist. And after 3 to 5 days, you have now memory B cells that are differentiated into antibody secreting cells. And now you can seed them into the fluorospot plate here. In order to detect if you have antigen-specific B cells, you have 2 options. The direct method and the reverse method, depending on how you use your antigen of interest in order to detect the secreted antibody that is specific. Another option you have is if you want to detect the total IgG or in general IG, the total antibody that has been secreted. And you do that by, again, capturing the secreted antibody and have another anti-FC antibody here binding to a different epitope that will tell you the total of IG that was secreted. I want to go a little bit deeper on the direct B-cell fluorospot. So here you would have your PBDF membrane coated, instead of an antibody as we saw previously with T cells, here you coat it with an antigen. So you coat it with the antigen of interest, you allow your B cells to secrete the specific antibodies, and then you detect that by using anti-IgG, IgM, etc. detection antibodies that are tagged or biotinylated. And then as a second step, you have the fluorophore conjugates or anti-tag antibodies to detect your specific antibody that are secreted. This is a case study where they use this method exactly. So here they had a cohort of about 20 individuals that were hospitalized for COVID-19, and they were interested in studying the B-cell responses from SARS-CoV-2. So this is a very good example of how you can not just use fluorospot, but you can complement different methods by using fluorospot. So here they used ELISA for assessing neutralizing antibodies specific for SARS-CoV-2. They also use flow cytometry to detect the antibody secreting cells by phenotype. And they also did an IgG, IgA, and IgM three-color fluorospot to assess the specific antibody secreting cells. By serology, they found that only 16 out of the 20 patients were seropositive for antibodies for SARS-CoV-2. Whereas for flow cytometry, they found that all of the patients actually had antibody-secreting cells. This is just general antibody-secreting cells, not against the SARS-CoV-2. So they wanted to take this a step further to see if this antibody-secreting cells was against SARS-CoV-2. They did this three-color fluorospot where they coated with a nucleocapsid protein, the membrane. They incubated the cells, allowed them to secrete the antibodies, and captured them. And this is what they saw. They saw that all of the patients actually had responses and had specific memory B cells in comparison to the healthy control. So what they were able to say here was that if they were relying only to ELISA, they wouldn't be able to see this. But because of the sensitivity of fluorospot, they were able to tell that actually all of the patients had B cell response. And also this was a very nice way to complement their other findings with flow cytometry and with ELISA in order to make their point stronger. There are many other potential applications of this method. For instance, you could test for cross-reactivity or identification of immunodominant viral proteins. With a reverse B-cell fluorospot, now you can use many more antigens, so up to 4. So here it's a bit different that you coat your PBDF membrane with a generic anti-FC IG. So for instance, now you can capture all your IgGs that are secreted. Again, you allow your B cell to secrete the antibodies. And now you have biotinylated or tagged antigens that bind to the secreted antibodies. So you have up to 4. Here's an example of just 3. And these antigens are tagged or biotinylated. So they bind to the antibodies that are specific to, and then you have the detection step, the second detection step with the fluorophores. This is a very nice study that used this method and actually developed to see if they were able to study malaria. So here they wanted to assess memory B cell responses in malaria newly infected individuals. They had a cohort of 20 travelers with acute malaria, and they took different samples at 4 to 6 time points over a year. And they wanted to see how the memory B cell response would change over time, so to immunomonitor this disease. They recombinantly produced antigens, malaria antigens that were tagged or biotinylated, and they did this reverse B cell fluorospot to assess the response. And this is what they found. In the acute stage, obviously there are no memory B cell responses, so they couldn't find any. But as the time went by, they were able to say that actually this particular patient developed memory B-cell response against 2 out of the 4 antigens. So they were able to study this and see which antigens were more relative and the more immunogenic in order to use them for developing different immunotherapeutics or vaccines. So to conclude, this is a case where they developed the multiplex fluorospot assay themselves. They were able to tell the heterogeneity of the memory B-cell responses. They were able to tell the differences between the different individuals and how the memory B-cell responses differed. And they were also able to tell how the antigen-specific immune response was maintained over time. Other applications here would be to immunomonitor an infectious disease, or to study the acquisition and maintenance of the memory B-cell response, or you could also study antibody cross-reactivity against different antigens. I mentioned a few of the applications, but there are many more, such as in allergy, autoimmune diseases, transplantation, different cell therapies, and different, of course, infectious diseases such as tuberculosis or HIV. We also have a lot of antibodies for veterinary research, so people can use them if they are developing vaccines for animals, for instance, or diagnostics. And this is the last slide where I just sum up everything we discussed. Basically, this is a multiplexed assay that you can assess analyte secretion of up to 4 analytes. You can assess different immunotherapeutics by using it, and you can definitely assess polyfunctional cells. By using the RSV, you can have a different layer of data where you can dig deeper into them and see the relative amount of the secreted analyte that you had in your assay. And with my thank you slides, I also would like to invite you to register to our free webinars. We are planning 2 at the moment. We have one on the 28th of May about B-cell profiling with ELISpot and fluorospot. And then on June 11th, we have another webinar for fluorospot in cell and gene therapy, where we go really in depth into that. And yes, this is my last slide. Thank you very much. Any questions?
Yes. Thank you for the talk. This RSV algorithm would allow us to compare data run in different places or different days, or is it too variable?
Yes, so with fluorospot you can. With ELISpot, because RSV is also there, you cannot do it with ELISpot, just because the development of the substrate can make your results differ. But because it's fluorescent signal with fluorospot, you can definitely do that.
Okay, thank you.
Any other question? Okay, thank you very much.
¿Hago yo? Sí.
Sí. ¿No hay de nada?
No hay de eso.
Okay, so now we move to the next session. The session 2, the NAИVE immunology session. So we have Javier to present his talk about CLL PDX models, right?
Yes. Sí, sí, a ver. Okay, so good afternoon. First of all, I would like to thank the invitation to get here today in the SCI. So I'm going to explain a little bit our projects in CLL and Richter transformation. So CLL is a malignant disease that is based on accumulation of mature B cells in blood, lymph node, and bone marrow. And in some cases, it has been described that CLL cell can transform into Richter transformation that is like a diffuse large B-cell lymphoma. So clinical data shows that more or less it is the overall survival of CLL patients, but if we compare with the Richter transformation patients, his overall survival is less from 1 year. So nowadays it's a very aggressive disease that there is no cure since now for these patients. So it has been described that Richter transformation patients had a higher genomic complexity, and also some mutations are shared from the CLL sample, such as, TP53, Notch, or SF3B1, and other alterations more regarding to the more proliferative status and all the activation of the cells, such as deletion in CDKN2A and B and MYC activation. So it has been described that the CLL patient, when it starts the transformation into a Richter, there is more DNA damage and more MYC activation that leads to immune evasion, cell cycle dysregulation, inflammation, chromatin remodeling, and other things. So with this information, we have published these 2 papers that describe how the Richter transformation cells are seeded at the beginning of the disease, and how, using a PDX model, we were able to recapitulate the transformation and look for new therapeutic options. So the first question was when these RT subclones emerge in the patient and starts the transformation. To do that, we select 19 cases that were diagnosed from a CLL that finally transformed into a Richter, and we took several time points from diagnosis until the transformation and several hits of the patient after different treatments. With all this data, we performed genome studies, transcriptome, and epigenome studies. So I'm going to focus in the first part of the genome studies. So by whole genome, we were able to infer the clonal evolution that took part in the patient since the diagnosis until the transformation. And as you can observe by bioinformatician analysis, we were able to infer that these subclones that goes up and down depending on the treatment were able to be inferred at the beginning of the disease. And finally, as you can observe here, the dark blue subclone, that is the ones that expand in Richter transformation, we were able to infer that is present at the beginning when patient is diagnosed from a CLL. Also, by single-cell DNA, we were able to detect that this transformation subclone has this specific mutation of TP53. So by single cell, we were able to detect that at the transformation, almost all the cells carry this mutation of TP53. But if we were back in the several time points, we were able to infer that the diagnosed at 0.09% of cells are carrying this mutation, meaning that this transformation must be at the beginning. Later, by transcriptomic analysis, we were able to dissect a specific signature that was characteristic for these Richter transformation samples that was not present in the CLL samples. Here are 2 specific Richter transformation patients that were specific that do not carry the signature. Because first, this case was a prolymphocytic transformation, not a Richter transformation. And the second one was a special case that, although was a Richter transformation, carries specific mutation in the IgL light chain of the immunoglobulins, meaning that there is a homotypic cross-link about all the both immunoglobulins, and that makes an autonomous BCR activation without an antigen. So we're able to see that this case activates the BCR activity, so cells can start proliferating without any type of antigen. If we perform a gene set enrichment analysis, and we're able to dissect from all the signature an increase of OXPHOS, that is the oxidative phosphorylation, a specific signature where cells start increasing all the metabolism of mitochondrial, and also at the same time a decrease on BCR pathway. That is the main pathway for B cells in order to start proliferating and induce the leukemia. So with this data, we were wondering how we can validate this functionally, the increase on OXPHOS and the decrease on BCR. So to do that, first we set up a new technique in the lab in flow cytometry in order to analyze how the BCR activity is in the B cells. So here is the pathway of the BCR. So when the BCR encounters an antigen, there is activation of this pathway through the BTK, and at certain point, this phosphatidylene biphosphate can go into deacylglycerol and isotyl 3-phosphate that goes to the endoplasmatic reticulum, and calcium is released into the cytoplasm. So we set up a technique in flow cytometry that we were able to detect this release of calcium in the cytoplasm. So an increase on cytoplasm in the cells means a direct activity of the BCR. So to do that, first we set up with Indo, that is a fluorochrome that enters into the cell and can catch the calcium. So when Indo catches the calcium, their structure is changed, so the emission of the light changed from blue to violet. So as you can observe here, this is the basal levels of calcium of the cell, and after incubation 2 minutes with an anti-IgM and hydrogen peroxide in order to inhibit all the phosphatase of the of the pathway, we were able to see that there is an increase on the ratio violet-blue, meaning that there is an increase on calcium in the cells. So with this technique, we were able to see that actually this BCR activity is or not working in our cells. So here is a first example of case 12 that had this BCR low signature in Richter transformation cells. And as you can observe here, the gray line that is the CLL sample clearly exhibits an increase on this ratio, meaning that there is activity on the BCR. Whereas when we analyze the Richter transformation sample that was BCR low, we were able to detect a small increase on the calcium influx, meaning that this BCR activity actually was lower compared to the parental CLL sample. Also, this activity was observed in this case 63, although was very variability in the clusters. So some clusters were BCR low and other was BCR normal, in both cases in the CLL or the Richter. So we were able to observe this decrease in the curve and were not maintained during the time. The second part was how to study the oxygen consumption of the cells. So as OXPHOS is a mitochondrial chain characterization, because it has been described that these cells increase their oxygen consumption from the mitochondrial chain, we analyze using this machine that is Ouroboros how the mitochondrial chain of the cells works. So first, we read the routine level respiration that all the mitochondrial chain is completely working. After that, we added several hits in order to block this mitochondrial chain and see how this was modulated. So first, we added oligomycin that blocks the ATP synthetase. So all the chemiosmosis of the mitochondrial chain was completely blocked. After that, we added FCCP, that is ionophore, that deregulates the inner membrane, meaning that although in the first part we block the ATP synthetase and all the protons are retained in the intermembrane space, when we add here the FCCP, we dysregulate this, meaning that the protons can recirculate and again, cells can consume oxygen. Later, we added rotenone that blocks completely the first complex, so all the mitochondrial chain is completely blocked. And finally, we added anti-mycin A to completely block complex number 4, that is the ones that actually use the oxygen. So what we are able to observe is that at the routine level, Richter transformation cells had more oxygen consumption compared to the CLL cells. So we validate this OXPHOS high profile that were observed using the transcriptomic analysis. When we added the oligomycin, all this oxygen consumption was reduced in all the cells. And our surprise was here at the ATC, that is when we added the FCCP, so the recirculation of the protons gets activated again, that we were able to observe that Richter transformation cells actually can consume more oxygen than the oxygen that they consume at routine level. So it seems that they are not at their maximum capacity of the mitochondrial oxygen consumption. Also, this study was observed in the case 63, again at the routine level, Richter transformation cells, we confirmed that they were OXPHOS high compared to the CLL, and also at their maximum capacity, these cells can go further and consume more oxygen. So with all this data that we confirm that actually these Richter transformation cells are already beginning of the disease and has this specific profile of BCR low and OXPHOS high, we generate a PDX model that is a mouse model that actually can recapitulate the transformation of the disease into a Richter in order to see which are the different mutations that are acquired in the cells in order to induce the transformation. To do that, we took blood samples from patients and we divided B cells from T cells. We stimulated by separate and then we injected into NSG mice. We use NSG mice because it's immune deficient mice, and in that case, we cannot get any allogenic rejection from the mice from our samples. After several passages, we were able to achieve 2 PDX models. In most of the cases, after an injection, we didn't get any type of engraftment, and in 2 cases, as you can observe here, we obtained 2 different PDXs, one that actually were at the subcutaneous level, that where we injected the samples, but when we performed clonality studies, we observed that was a different clone. And this is why, because both patients were infected with EBV virus, and EBV induced proliferative status in B cells. And these 2, we found that although we injected subcutaneous, both cases were infiltrated in the spleen. And for this case, that actually it was an infiltration of the B cells. The infiltration was again from a different different clone and were again EBV positive. Finally, 2 PDXs were well established and one come from a Richter transformation sample. So we consolidate the transformation from the patient. And the other case, we started with a CLL sample that finally we get a Richter transformation. So with this model, we were able to confirm that actually these RT subclones must be already at the beginning. If not, if we injected the CLL and the cells are not at the beginning, we are not able to induce the transformation in mice. So after these 2 cases, we injected endovenously in mice and we were able to observe that we can generate a leukemic mouse model that actually can recapitulate clearly the transformation of Richter in order to assess new potential therapeutic strategies. So both PDXs were validated first by flow cytometry. So as you can observe here in forward and side, PDX samples had the same profile of size and complexity compared to the Richter that are more larger and complex cells because are more similar to diffuse large B-cell lymphoma compared to CLL cells. And in both cases, maintain the main markers of CLL cells that are the CD19, a B-cell marker, and CD5. And regarding clonality studies, we were able to observe that at early passage and established passage, the clonal peak was the same as compared to the original sample, meaning that actually the clone that engraved in mice was the tumoral clone from the patient. So as you can observe here, that this is the clonal evolution that took part in the patient. We took this sample that was prior ibrutinib. Ibrutinib is a treatment on CLL patients that is a BTK inhibitor. So as you can observe here, when we inject this sample in mice, the clonal evolution that took part was very similar to the ones that took part in patient when transformed. For example, this pink subclone, that's the one that diminished during the transformation, and also in our mice was the same clone that diminished. And this subclone that was the subclone of Richter transformation, the dark pink that expand in the patient, also was the ones that expand in our mice. But also these results, we were able to infer that actually these Richter transformation subclones are very heterogeneous. As you can observe here, from this RT subclone arises another ones that induce more aggressiveness of the tumor. That this was not be able to be observed in the first study because as patients die very early and we cannot collect samples after the transformation, the bioinformatic tools cannot infer this heterogeneity inside the Richter transformation subclone. So again, we performed this Richter transformation signature, and we were able to observe that our PDX samples that are the dark blue here exhibit clearly the Richter transformation signature compared to the original patient. And again, in this Richter transformation signature, we observe the increase on OXPHOS and again the decrease on BCR signaling. So we are able to recapitulate the transformation that occurs on patient. Again, we performed the flow cytometric analysis in order to confirm this decrease on calcium influx, so a decrease on BCR signaling. And again, the dark blue line that corresponds to the PDX will match with the Richter transformation from the patient regarding the calcium influx and were lower compared to the CLL sample. So we are able to confirm that actually this BCR low OXPHOS high profile is not a profile that was induced because patient was treated with one drug or another, and actually is indeed in the transformation. So the Richter transformation subclones already carry this signature that can be specific for these Richter transformation clones and can be used for therapeutic strategies that I'm going to show you now. Again, regarding the OXPHOS profile, our transcriptomic our Ouroboros analysis show that Richter transformation had more oxygen consumption compared to the CLR and comparable to the Richter of the patient. And again, at their maximum capacity, these cells can consume more oxygen. So with all this data, we wanted to assess new therapeutic strategies. The first one, it was described that there is a drug that is called Ajax that actually can target the complex one of the mitochondrial chain. And in that case, we can block completely all the OXPHOS profile. And as these Richter transformation cells have this OXPHOS high profile, can be a specific treatment for all these Richter transformation that do not affect other cells. So as you can observe here, the non-treated cells, when we added the FCCP and we induced the maximum capacity of oxygen consumption, reaches their maximum capacity compared to the treated cells with Ajax that we were not able to induce more oxygen consumption. So it seems that we can block completely the mitochondrial chain. This data was then validated by proliferation assays using CFSE in flow cytometric analysis. As you can observe here, first of all, Richter transformation cells show higher proliferation capacity compared to the CLL cells, that this concords with the MYC amplification that has these cells, that is a proliferative marker. So as you can observe here, when we treat cells, only those that had this OXPHOS high profile, we were able to reduce their capacity of proliferation, and not those cells that do not have this OXPHOS high profile. So we confirm that these Ajax drugs can be specific for these cells that are OXPHOS high. Later, it has been described in acute myeloid leukemia that when you combine this OXPHOS inhibitor with venetoclax, that is a BCL2 inhibitor, we can circumvent the venetoclax resistance, because it has been described that most of the patients on CLL are resistant to this BCL2 inhibitor. So as you can observe here, when we treat our PDX with venetoclax, that is this BCL2 inhibitor, we are not able to reduce the viability of the cells in either both cases. But when we add the combination of venetoclax with Ajax, we are able to reduce the viability with a synergistic effect in both cases. These results in vitro were also observable in the in vivo studies. So as you can observe here, when we combine venetoclax with Ajax, we were able to observe that were the mice that has less spleen infiltration. And finally, the readout that was the relative spleen weight, we were able to observe that the combination was the ones that get less spleen infiltration. So with all this data, nowadays we are trying to assess CAR-T therapy in order to see if actually we can circumvent, apart from this resistance, the resistance that it has been shown that Richter transformation cells can regulate the CD19 and escape from, for example, in that case that we are working in hospital clinic, the treatment of RE001, that is an anti-CD19 CAR-T therapy. And with that, I would like to thank all the members from IDIBAPS and also IDIBAPS and Barcelona Supercomputer and all the funders. And if you have any question, I will be glad to answer you. Thank you.
So any question?
So I'm wondering about the BCR in this transformed subclones. So is there a problem with expression on the cell surface? And linked to that, I saw that for the calcium studies, you use anti-IgM. So I assume that all express IgM, but do some also express IgD? Or do you have some even that express IgG? Or so my question is, what happens with the BCR itself? Have you checked if there is a deficiency in expression or another issue with that?
Yes, actually, by RNA-seq, we detect that is a low expression of mRNA of the BCR, and also by Western blot, there is a decrease on the protein. So actually, there is a decrease on the BCR signaling because there is a decrease on the protein, because these cells actually induce another way to proliferate, because BCR is the main proliferation of the B cells. So they activate mTOR, disoxphos, and MYC. So they go to another way, because as we are using BTK inhibitors, it has been described that this subclone escapes from the treatment and there is a decrease. And regarding the IgM or IgD, there is a paper that it was published that says that in these leukemic cells, most of the cells, about 90% of the surface IGs are IgM instead of IgD. So it seems that IgD is not affecting on the proliferation of the BCR. So we go further to the anti-IgM and we are not going to detect the IgD. So it was based on literature in that case.
And just a comment, I mean, the in vivo data, the model, that's amazing, because this is extremely difficult to reach those. So those results are really impressive.
Yeah, exactly. Thank you.
Any more questions? I have a question for you.
Yes.
Because I saw many of your CLL samples didn't engraft in the mice.
Yes.
So you are transplanting them subcutaneously.
Exactly.
Is there any possibility to transplant in any other more favorable microenvironment for T cells?
Yes, actually, there is a paper that we based the work that they try subcutaneous and intraperitoneally. The thing is that when we inject it intraperitoneally, most of the cells spread to the spleen, and we injected both B-activated and T-activated cells. And when the cells goes to the spleen, T-activated starts proliferating faster than the B cells. So there is a huge, massive invasion of the T cells. So you lost all the tumor. Whereas when we injected subcutaneous with matrigel, you encapsulate all the tumor there. So there is angiogenesis over there. And we observed that after several passage, only CD4 remains. That seems that helpers to engraft the tumor. And then when the tumor is engrafted and can proliferate alone, all the T cells go back, and only the tumor is on the subcutaneous niche. So, but as you can observe, only 2 out of 11 we engraft. And we observe that because we try CLL patients without the transformation, and actually only the transformated patients that are more aggressive actually engraft. So it seems that maybe this aggressiveness induced the engraftment in the mice. And some mutations that we observe, that is SF31 or NORCH1, are specific in both PDX. So maybe there are also specific mutations that also induce the engraftment. Yeah. So we are now studying about that.
Okay, thank you.
You're welcome.
Okay, if there is no more questions, we can move to our next speaker. So, Macedonia Trigueros from IrsiCaixa. She's gonna tell us about spectrum flow fish.
Okay, thank you for the presentation. This talk is a little bit related with the talk of Marco, because we developed this protocol together. It's a multiparametric characterization of telomere length in T cells of people living with HIV using full spectrum cytometry. Well, for give you a little bit of background, I am doing my PhD in immunoaging, that is the change that take place in our immune system over the years when we are getting older. And this change will have an effect on our health and organ system, that is the development of comorbidities. So, but what happened with the immunoaging in people with HIV, where there are two key points here. The first one is that people living with HIV present a higher proportion of comorbidity. And the second one is that these comorbidities appears at younger age. So what happened? Why happened? What is the reason? Well, there are different hypotheses or factors that can affect to this increased comorbidities, that these people is usually under antiretroviral therapy. Also, they used to have a persistent inflammation and immune dysfunction, and between other factors. So we hypothesize that the increased comorbidities that we observe in people living with HIV is associated with some of these factors. We will see in these people a higher activation, immunosenescence, telomere shortening, and inflammation. So with this hypothesis, we want to study the telomeric shortening in the different T cell subpopulation from people living with HIV and compared with an infected population. So at the beginning of the studies, we know that we want to study the T cell because, as you know, CD4 T cells are the main target of HIV. And also study the distribution of the subset can give you an idea of the aging status of a participant. Also, we think that it could be interesting to know the status at immune status markers, such as exhaustion, senescence, and activation. And finally, we want to add to our studies a molecular marker that is the telomeric shortening. And also we want to measure all these parameters in the same sample at the same time. Obviously, we have the limitation of the number of PBMCs available. So, as you know, the telomere shortening is this process is normal, is related with aging. So we can measure the telomere shortening in different populations by a classical approach when we have to split our cells by magnetic separation or shortening, and follow it by a second step of telomeric measurement that could be by Schiller-Blanc or qPCR. So, but for these techniques, we have to split our cells. We cannot measure all in the same sample. So, another nice approach is this fish technique. When we have ordinia, we are to denaturalize and we add a proof for measure the telomeric size. So we can measure, in this case, the telomere by microscopy or by flow cytometry. There are many papers where they explore the flow fish. Well, and some advantage of use flow fish is that we not need cell shortening, we not measure subtelomeric linear regions, or it's require less cells, and we can measure membrane cell markers and telomeric shortening in the same experiments. So one thing to take into account is that when we measure telomeres, or when we want to develop a flow fish, we have to add a cell cycle staining instead of life and death. Why? Because we want to measure telomeric shortening, but just in the cells that are not dividing, because in the cells that are dividing, that are at least, we can increase the amount of dynia if this will increase the signal of telomeres. So a typical protocol by flow fish have this first step when we develop the cell surface staining with the fluorochromes. We did the first fixation, and in the second step, we developed the telomere staining. But as Marco commented this morning, we have this denaturation and this bot fixation that is a little bit aggressive with our fluorochromes. So let me explain what was the challenge of develop this flow fish. The first challenge is this: not all the fluorochromes will tolerate this protocol. And the second one is that when we are use cell cycle dye, this cell cycle dye used to be very bright signal. So this will introduce spreading in our data. And also, when we develop flow fish, we had this increase of autofluorescence that Marco commented this morning. So, as you can imagine, to design a panel for flow fish is not easy. So, that we have developed is a new technique to measure the telomeric telomere led by flow fish using full spectrum flow cytometry. We are starting at the beginning of the experiment. We have a normal panel with conventional cytometry when we have 8 markers, 8 fluorochromes, sorry, and we are able to differentiate these different T cells population as NSM. But when we move to full spectrum flow cytometry, we are able to increase the number of markers. And obviously, we increase the ability to measure marker of status and increase the number of population that we are studying. So for give you a little bit of how difficult was develop this panel, this is an example of a 40 color panel that Marco told this morning. This is a well-characterized panel. This is Omic 69 that works very well for our panel. So if we want to have the crazy idea to develop flowfish in this panel, what could happen? Well, the first of all is that, as I explained before, not all the fluorochromes resist to the technique. Here, I show you, well, in gray is when we do the normal staining without fish, and in red when we develop the fish protocol. As you can see here, there is no signal, for example. So, where there are other fluorochromes, polymer-based diet that work well. So, this is the first challenge. Not all the fluorochromes available can be used in this methodology. So remember that we have 40 panels. After this limitation, we have 50 fluorochromes that are not able to use in our flowfish technique. So another aspect to take into account is that remember that we are a cell-cycling dye instead of life and death. So sorry, instead of viability. So we started testing this, but if you see here, the Vibram ruby is cited. Yes, is cited by all the laser, and this is the one we can see here is spillover that we'll be introducing in the fluorochromes that we have here. No, and what fluorochromes are located in these regions? The fluorochromes that are able to tolerate the protocol. So we were looking for another cell cycle diet, and we found this one that, well, we have some problems that we solve it with higher dilution. And finally, we can use this for the cell cycle diet. Well, this is our final panel design. As Marco commented this morning, full spectrum flow cytometry allows use combine fluorochromes that normally in conventional are not possible. Also, this is your diapositive is your slide. As Marco explained this morning, with the fish, we increase the autofluorescence. But when we are used, in this case, Aurora, is mean the full spectrum flow cytometry, we are able to develop autofluorescence station automatically. So this improve the resolution. Well, this is the final panel that we have. This panel allows you to detect different population. Well, our hypothesis that when we are younger, the proportion of naive is increased. In baby, for example, have a high proportion of naive. But when you are getting older, this changes, and your cells are more differentiated, and you have more this phenotype, no? So also similar happen with the telomere size, no? When you are younger, you are longer telomeres than when you are older. So for measure the telomeric size, we have this cell line that is commercial cell line with highly conserved telomeric sequence, and we use it as internal control for measure the telomere length. And well, basically this is the protocol. We have to do duplicates. In both we did the extracellular staining, okay, but just in one we have the proof. So we have here the demo, but just here the proof. And the second day, well, we add the DNA staining, and well, we will to our graph. This is the typical gating that we develop. This is the cell line that we are using for measure the telomere size, and this is the lymphocyte of the different participants. Well, with the cell cycle dye that we are using allows you to separate the cells that are not dividing. Finally, we are able to identify all these populations. And additionally to all these populations, we're able to measure the status of all these populations using these immune system status markers in all these subpopulations. No? We measure senescence, exhaustion, and activation. For measure the telomeric, really that we did is a relative telomere length, when we have to take into account the MFI of the population of interest and the MFI of the internal control. So, after this we have, this protocol works, is okay, or so here I show you some examples of validation. The first example is that we are, when people is younger, have no expression of these markers that are markers of senescence. When you are getting older, you start to express this one, and when you are more older, you express both. So if we look at the telomeric shortening in the different T cells expressing this or not expressing these markers, we can see that the higher expression of senescent markers correlates with shorter relative telomere length. So another validation is in this case, when we have naive central memory, theoretically we have increased telomere size in this T cells subpopulation, and we can verify this in the CD4 T cells and CD8 T cells. So we conclude that our protocol works well. Now I show you so quickly the results of our mice studies using this protocol in patients. So we have over 50 cohorts. That is curve integrates for people with HIV, but under antiretroviral therapy. And also we have HIV negative control. The people with HIV used to have increased comorbidity, as I explained before. So, when we move to the results, we can see the people living with HIV used to have a lower proportion of CD4 T cells. That is so typical. No difference were found in the distribution of the T cell subpopulation. Regarding to the telomere length, we found some tendencies, but that people living with HIV used to tend to have a shorter telomere length. When we move to CD8 T cells, we found that people living with HIV have increased proportion of CD8 T cells. No difference were found in the distribution, but we found that regarding to telomere length, people living with HIV used to have shorter telomere in naive central memory and transitional memory substance. And regarding to the cell population, we find a little bit more activation in CD4 of people living with HIV. And regarding to telomere length, we just find that the exhausted CD8 T cells of people living with HIV used to have shorter relative telomere size. So just to finish, I would like to conclude that flow-fish technique is using full spectrum flow cytometry, allow use a depth immunocharacterization of the relative length in CD8 and CD4 T cells in people living with HIV and in the infected controls. And to give you an idea of our experiments in number, we have just 18 markers. No 40 like this morning explained marker, but with these markers we are able to identify 15 T cell subpopulation. We are able also to identify in each population 5 immunomarkers of status. And finally, we are able to measure the telomeric size, not just in general population. We are able to identify the telomere shortening, for example, in NAIVE that are expressing markers of activation. So regarding to my results, we conclude saying that despite this powerful technique, we observe a difference in the relative telomere length between people living with HIV and uninfected controls. So our results suggest a similar immunoaging status in older individuals, regardless of HIV serostatus. And our next step, future purposes, we are developing a new panel when we are increasing the cohort adding younger age, and also we are increasing with three, 4 markers. It's at more is a little bit complicated for understand what's happening in the immunoaging of HIV people. So thank you to my team, especially to my boss that is here, Marta, especially also to Marco and Hamad that helped me to develop all this protocol. It was no easy task. Thank you so much for your attention.
Thank you for your presentation. So is there any question?
So, I have a question. So it's a kind of technical question. So you studied how the treatment affects the different fluorochromos, but is it the possibility also that the treatment also affects some of the cell surface molecules, and by affecting the molecule, then the antibody gets detached from the cell surface? Or it's just the fluorochromes that you should worry about?
Well, it could be a fit, but for this reason, we have a first fixation. When we add the marker of membrane, we add the first fixation. I understand that in part is for avoid this, but
So, the fixation include the fixation of the antigen antibody. But at the beginning of this protocol, we compare the same expression in fresh sample without any pattern of the expression with the final pattern in the after the two fixation and the 22 degrees. So there is no the key of this protocol is the first fixation. Because this fixation stabilizes
the membrane, no? The membrane markers.
Phenotyping.
More questions? Do you think this telomere shortening is specific of infection of HIV, or any other inflammation process or chronic infection can also have same effect?
Yes, also I know that, for example, the infection with cytomegalovirus, no, is to be correlated also with the telomeric shortening, but there are many factors, no, that can. It's complicated. It's not easy, no, to say it's just this factor or this one. Just.
Okay. Thank you, Macedonia. So let's move to the
So we still have a session.
Sí, que ahora voy al hospital.
I don't know if the speaker is here. Oh, yeah. Paula?
Yeah.
Okay, so yeah, you have to upload the.
Thank you. Sí, mucho labor, sí. Es una experiencia. Pues yo con la máquina dice.
And wait until 30 or I can wait, no problem.
Sí, sí, yo soy sí,
No problema. Okay, so we're going to start with the last presentation. We left the most interesting one for the last, but we lost some of the students, I guess. So Pablo Guerra, he's going to talk about a very interesting technique with his cryo-IM, and then you will talk about what can do cryo-IM for us. Yeah, okay. Thank you so much for accepting our invitation.
Thank you. Hello, everyone. Basically, today I'm going to give you a brief description about what is electron microscopy and what can you do in life science with this technique, okay? Basically, the idea is that you can use a microscope that uses electrons instead of light to imagine your proteins or your cells. After that, you can put all these images together and you can obtain a three-dimensional reconstruction of this protein, purified or in cell arriving to high resolution. You can see the position of side chains, all the atoms in complexes that you are looking for. But why there are people interested in know this information? The thing is that there is a lot of protein inside the cell, carry on a lot of different functions. And the function of a protein is directly related with the structure, with the three-dimensional structure of this protein. If you know how this protein is, you can know how this protein is going to carry out the function. So this is a very powerful information in the case that you want to improve the function of a protein or do you want to stop the infection of a virus, for example. So one example. Hemoglobin is the protein that take the oxygen from the lungs and brings to the rest of the body. So how this protein is able to do it in a reversible way? In 1956, I think, Max Perutz saw the structure of this protein of hemoglobin using X-ray diffraction. And now that this protein is formed by 4 monomers, and each one of them has an iron atom in this region that are able to bind the oxygen molecules in a reversible way. Knowing the structure of this protein, this guy was able to know how this protein is able to carry out the function, okay? That's the idea. Depending on which one is your biological question, you have different techniques. In the case of electron microscope, historically was situated between the light microscope and the high resolution techniques. But in the last 10 years, there is a lot of improvements in hardware and software that we talk later that can use this technique to arrive to high resolution to see the position of all the atoms of your protein, okay? Okay, this is a microscope and it's not far away from a light microscope. Basically, the idea is the same. You have the same elements. You have a source, you have lenses. You are going to be a detector here to obtain an image of your protein. The difference is that you are not using light, you are using electrons. So you need an electron source, obviously, And instead of using the typical optical lenses, we are going to use electromagnetic lenses to focus the electron beam and applying over your sample, and again applying over your detector to obtain this image, okay? When we talk about electron microscope, we have to keep in mind that there are two different electron microscope techniques. There is a transmission electron microscope, and also there are the scanning electron microscope. In both of them, we are using electrons to imagine our sample, but we are going to measure different electrons. In the case of the transmission electron microscope, we have our electron beam that is going to arrive to our specimen, pass through, the electron beams pass through your sample, and are going to obtain an image, okay? This is transmission because we are going to measure the electrons that are transmitted through your samples, okay? In the case of a scanning electron microscope, we are going to beam of electrons that are going to arrive to your sample, but we are not going to measure the electrons that are going transmitted. You are going to measure the electrons that are diffracted, okay? And this is the same thing. This is a pollen grain, and this is the picture that you obtain using a transmission electron microscope that looks like a two-dimensional projection or slice of your specimen. And this is the typical images, beautiful ones that you obtain with the scanning electron microscope with shadows that looks like more a three-dimensional reconstruction, okay? So today I'm going to talk only about transmission electron microscope. I might also say that we are cryo electron microscope. Why I say cryo? The life science samples like proteins in solution or cells are water samples. So if you put in an electron microscope, the radiation damage that this sample receives is too high that you need to protect the samples. And how we can protect the samples? We need to freeze our samples. That is that you have your virus in this case, you are going to freeze to protect from the radiation damage of the beam, okay? With this purpose, we use this thing that is called a Vitrobot that I show you later. Okay, just to introduce 4 slides of history. In the 90s, at the beginning of the last century, the light microscopy was arrived to the limit in resolution, but there are a lot of people that wants to know how are a protein, how is a cell with more detail. So basically, Ernest Rusk and Max Wall are going to think about it, how we can obtain this information. And we see that the electrons has properties that are very good to obtain information at high resolution for the wavelength and blah, blah, blah. So putting all this information together, they are able to buy at the first transmission electron microscope, and the next year, the first scanning electron microscope in 1936 or something like that. They are going to receive the Nobel Prize for this discovery. But yeah, after that, Leslie and Martin say, okay, this stuff is really good, but you cannot use for life science samples because the electron beam is so hard, the radiation damage is so powerful that are going to destroy everything that you are going to put it inside this microscope. And give some ideas about how we can protect these samples to put inside the microscope. That's the first ideas about negative staining approximation and the cryo-electron microscopy. One thing that we have to keep in mind also is that we have our microscope, okay? We have the column and a beam of electrons. This beam of electrons are going to interact with your samples, but also are going to interact with everything that is in the air. All the molecules that are in there are going to disturb with your electron beams and are going to increase the noise of your images. So one thing that is very important in all the electron microscope is that there is a very high vacuum inside the microscope, okay? To remove all the particles from the air and avoid the interaction between the beam and all these molecules that are in the air, okay? This is another thing that we have to keep in mind because our solutions are in water, and if you put water inside a vacuum system, it's going out, okay? So apart from the idea to use cryo and to beautify is not only to protect from the radiation damage, it's also to dry the samples, to move to solid state, okay? To be able to put inside the microscope. So with this purpose, in 1986 or something like that, Jan Dubochet is going to develop the the strategy that we use to use cryo-electron microscopy nowadays. That basically the idea is, okay, you have your cells, or you have your protein in solution, you are going to put it over a metal piece, introduce in an ethane as fast as you can. Why? Because we need to avoid the crystallization of the water. We need to vitrify because the crystals looks like black. So faster you vitrify, faster you freeze, you are avoiding the formation of this kind of stuff, okay? So, okay, this is like a guillotina, yeah? That I'm going to introduce sample is an ethane at minus 196 degrees, very fast to obtain the sample to put inside the microscope. This guy is going to receive the Nobel Prize also. And finally, Joachim Frank and Richard Henderson are able to manage or to describe how to manage these very noisy images, because when you see the protein, for example, in this case, or cells in the electron microscope, the signal is very, very low. The noise is very high, your signal is very low, so it's hard to see something. So this guy and this guy are able to work together to try to improve the signal of the protein or the cells to be able to solve the structure of three-dimensional at high resolution, okay? Okay, when we talk about cryo-electron microscopy, you have like two different approximations. We can talk about single particle analysis, and we can talk about cryo-electron tomography. That's depend on the objective, but basically you are going to this way when you have small stuff like protein that you are biochemically purify, big macromolecules, blah, blah, blah. And in the case that you are working with big stuff like cells or tissues or organs or something like that, you have to move to cryo-electron tomography, okay? First approximation, single particle analysis. Basically here we have protein in solution, complex with DNA, complex with the IgG, whatever. You are going to vitrify, you are going to put inside the microscope, you are going to acquire a lot of images of the same protein in different orientations, and you are put all this information together to obtain a three-dimensional reconstruction of this complex that you are interested in to know, to describe the thing that is going on. So this is the workflow. This is just to say you one idea is that this is the first step. This is the high resolution reconstruction. You are going step by step, but in any time or in any step of this cryo-EM workflow, you have to come back maybe to the first, if something is going bad, maybe you have to come back to this step, or maybe you have to come back to the beginning, because it's not so easy to arrive to high resolution. In the case that you need to know the position of each atom, you have to work very hard and you have to get a very, very good sample, okay? The first thing is our sample that we describe that this is a liquid protein in this case, and we are going to vitrify. We need to put it, we need to freeze, but we need a support. So basically the support that we are going to use is it's called grid. Basically it's a metal piece of circular shape where inside, when we made inside this, this grid, there are holes. And in these holes are going to put it your protein to obtain images, okay? If your protein is put it over the support, we cannot image. We need to take pictures only in these holes, okay? There are different size of holes, different materials, blah, 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 a lot of things that you can play. You try to increase the, it's pretty cool. If you have any question, you can stop me, okay? That is, if anyway, I continue. Okay, the vitrification as we described before, the idea is that we have our solution, in this case a protein, and we need to vitrify, we need to freeze as fast as we can to avoid the crystallization, okay? Because the crystals of water are black in the microscope and you cannot see your protein. So basically the idea is you have your tweezers and you take your grid, your metal grid, you are going to apply the protein solution, and to obtain a very thin layer, you need to dry just before put it inside the ethane that is a minus 180 degrees, okay? We need to dry before to put it inside because the drop is too thick that if you introduce by itself, it's completely black. The electrons are not able to pass through the sample, okay? So this is the equipment that we use to vitrify. This is called vitrobot, that it's quite easy. It's a guillotine. Basically, here there is a chamber with the humidity and the temperature control. There is the tweezers with the grid. And in this region, we have the ethane at minus 180 degrees with liquid nitrogen. And there is a video that is easy to understand. This is the grid with the tweezer. You put it inside the chamber of the vitrobot. You are going to apply your protein, and now very fast are going to dry using this filter paper, and after that is going to put it inside the ethane. Well, don't worry, that is on. It's very fast, that's the idea, okay? Liquid nitrogen, blah, blah, blah. So yeah, you have your samples, you prepare maybe 10, maybe 20 different grids with your different complexes, blah, and you move to the screening microscope to see how it looks like your protein the first time, and you are waiting that when you move to a hole, because remember, we have to imagine that only in the holes, you see your proteins with different orientation, but in most of the cases, when you arrive to the microscope, most of the protein is out of the holes or is very close to the edge that was not able to process it, or you cannot able to obtain a reconstruction, or all the proteins are in the same orientation. That it is a problem. If you don't have all the information of the different orientation, after that, you are not able to obtain a three-dimensional reconstruction, okay? You have to start a work until you obtain something like that, okay? This is a proteasome when you are able to see that the particles are good distributed around the whole with different orientations, side view, top view, inclined one. So good starting point to move to the next step, that is the three-dimensional reconstruction, okay? When you have the gold grids, you have to move to a higher microscope to obtain several thousand, 10, 20,000 of images, put all together, running a very complicated processing, long in time, using GPU calculation until you obtain this reconstruction, okay? That I'm going to give you only some ideas to know how is this processing carried out, okay? The first thing is that we have to acquire a lot of pictures. Basically, this is the grid. And inside the grid, remember, there are holes, and we are going to image only in these holes. In the past, we have to do it by hand, but right now, there is good software that can do it for you, and you can send the targets from the different holes. You go home, and the microscope is going to collect the whole night until you have 3,000, 4,000, 5,000 pictures, sorry. That you go to the lab running a pre-processing that you want to worry about. But this is, for example, one example of images that you can obtain, okay? And remember that the signal of your protein, that is, for example, this is the protein, is very low compared with the noise that you have. And the software was not able to select only the proteins. So you have, by hand, you have to click in some of them, some of the, where you are, the proteins, you have to give to the protein, to the software, sorry, the coordinates of the different proteins, and after 20 or 30 pictures, the software are able to deal with the rest of the data set, okay? And after that, we are going to extract the particles. What that means? The idea is that you have your picture with a lot of particles in different orientations. If you are interested in the three-dimensional reconstruction of this protein, so you are going to cut all the different particles and you are going to put all this information together, okay? In the previous slide, we have a lot of different particles, and the idea here is, okay, we are going to cut any of this one and I'm going to put all together, okay? That's the extraction. And the good thing is that for any of these images, you have all the information of all the atoms of this protein, okay? You are not able to see, but the information is there. The only thing you have to do right now is you need to classify. You need to put all the different particles that are in the same orientation together, in the same group, in the same class, and all the signal is going to appear. All the information at high resolution, all the details start to appear when you are going to put all these particles together, because you are able to enhance the signal and fight against the noise, okay? The big noise that you have in your pictures. So, yeah. You are in situation after that, you got a very good subset of particles, different orientation, and you can move to the three-dimensional reconstruction, okay? The first thing, okay, you need an initial model that in the case that you have a structure of a protein you can use. You can use AlphaFold, different softwares that can give you an initial model with a very low resolution that you are going to use to classify your set of particles. For example, in this case, this is a classification with 6 classes, and 3 of them are rubbish, there are nothing, there's only noise. One of these, there are particles that are completely broken, and there are 2 very interesting. There is one particle, there is beautiful particles, the whole particle, and in this data set also was picking only half particle in solution. There is one thing that we are not looking for, but appear in the solution in this data set. So after that, you can extract this data set and using refinement, arrive to the high resolution, okay? In this case, you are able to see the side chains of the different monomers. This protein is called BOLD that appears in this slide, that it's forming by 78 copies of the same monomer, and how these monomers are interact with each other was described with this structure, okay? After that, because our result is a density map, it's not the typical colors. After that, the thing that we are trying to do is modeling that basically is like, you know, the residues of this protein, you are going to put it one by one inside this blue cloud to represent and be able more easy to describe the results that you are looking for. One example, in this processing that I will show you, the protein is called BOLD, and in this data set, we are able to isolate different conformation of the same protein, because one thing that is very important to keep in mind is that the protein is not a rigid thing. The protein is going to carry out a function, and during this function is going to change the shape, it's going to move, okay? So if you are able to isolate it, the different conformation changes that are going to occur in your protein, during the function, you are going to tell the story about how this protein is able to carry out. So in this case, the BOLD particles are involved in the transport inside the cell between the nucleus and the cell. And here we were able to see the different changes that occur in this particle in time that was triggered the opening of this particle in 2 halves to be able to encapsulate the drug or whatever and move to another region in the cell and again release there. Okay? Okay, single particle analysis finished. Cryo-electron tomography. I'm going to. The idea here is more or less similar. The thing is that we are using this approximation when we have big stuff, tissues. We are working with bacteria, whole bacteria, or cells, or we want to know this kind of information, okay? In this case, it's basically the idea is you are going to, it's better here. You have your sample, and if you remember in single particle, we are acquiring one picture in different holes, and we are going to acquire several thousand of pictures. In the case of cryo-electron tomography, basically we have our protein of our cell of interest, and we are going to acquire one picture here, and after that we are going to tilt 3 degrees or 6 degrees. We are going to acquire a next picture and tilt again, and after that in the other direction, and you are going to obtain a 3-dimensional reconstruction of the protein making tomography, okay? It's like in the hospital. So basically after that, you can put all this information together and obtain a 3-dimensional reconstruction of your whole cell, okay? Okay, the bottleneck or the main limitation of this technique is the sample preparation. It's not easy. In the case that organelles or cells that there are not too thick viruses, we can freeze our sample with the same vitro that I showed you before, okay? But when we are working about tissue and organisms, we have to move to high pressure freezing, that it's really a little bit complicated. And after that, we have to milling. We have to reduce the thickness of the sample. Why? Because if you put something that is thicker than 200 nanometers in the microscope, the electrons are not able to pass through, okay? So there are not anything, it's black. So you need to milling, and there are 2 different approximations. The fifth is the focus ion beam milling that basically is going to use a laser to remove all the cell and only obtain a small lamella with the region of interest. And another approximation is the cryo-ultramicrotomy, okay? Okay, when you have small bacteria or viruses or something like that, you can use the same vitro that I showed you before. That is exactly the same. You have the grid, you can grow the bacteria, you can put the bacteria over the grid, and after that, you are going to vitrify. In the case of bigger stuff, remember, we need to use the high pressure freezing. The basic idea is that you are going to apply very high pressure over your sample to move in the Le Chatelier diagram to move from the crystallization to the vitreous state of the water, okay? Don't worry about that, it's just different. When you have the thicker samples, like I explained before, we need to make thinner. We have to make milling. The old way to do it is the cryo-ultramicrotomy, that basically this is a diamond knife, okay? And here there is the pellet of your cells or your tissue or whatever, and you are going to cut. Using this diamond knife is going up and down, and are going to cut your pellet of cell or your tissue, obtaining very thin layers of cells that you are going to put over your grid, okay? After that, you take your grid, put it at the microscope, and you can see the cells or the, I don't know what is that, mitochondria, whatever. But there are bad things that you are introducing these artifacts. Can you see these lines that are producing this because the diamond knife is cutting the cells, okay? So maybe it's not the best way to obtain. This is a very smart way that is called the 5th-gen focus ion beam, that basically you have your cells with some fluorophores, so you can use cryocorrelative to see which region or which cell you are looking for or you are interested in, and uses this focus ion beam scanning electron microscopy. You are going to move to this cell and you are going to obtain a very thin layer. It's called lamella of the region of interest of your cell. You are going to remove all the rest of the cell above and below, and you are going to take this lamella to obtain a reconstruction, okay? More or less, that's the idea. The idea is that we cannot put a whole cell inside the microscope. That's impossible. You are not able to image something. You need to make thinner, and this is the best way that you can do it. Okay, you have your lamella of your region of interest close to the nuclear or whatever. And you remember we are going to image, and in this case, we are going to image in the same region, changing the angle. We are going to tilt the sample, and we are going to obtain a different set of images of the same body, of the same sample, changing the angle, and you are going to use this information to obtain the 3-dimensional reconstruction, okay? So one thing that we have to keep in mind is that we are going to tilt the sample, maybe this one. We are going to tilt the sample to obtain different images, but we cannot tilt the sample all that we want, like for example, in this way, because it's so thick again that the electrons are not able to pass through, okay? So historically, we cannot only tilt until the plus 70 until minus 70. This is a region that is called the missing wedge that you are not have any information, okay? And here, this is a picture of Einstein just to know how this information is going to affect to your reconstruction. This is how it looks like the same picture reconstruct, losing the information related with the missing wedge. So you are going to lose a lot of information. But this is a limitation that we're not able to solve. Okay, we are going to, you have to tilt series acquisition, and you are going to make the 3D reconstruction. You can do it in the real space, and you can do it in the fast Fourier space. Anyway, and this is the reconstruction that you obtain, okay? In the case of these cells, this is the nucleus. You have really a lot of information, actins, microtubules, ribosomes. So one thing that was very useful in tomogram is after you have your 3-dimensional tomogram reconstruct, you are going to segment in different regions. In the case that you are interested in going to the membrane, you are going to cut this tomogram to see the details in this region or in this region, just because trying to manage this big information is impossible. You need to focus, okay? And in the case that you are lucky and you have a lot of protein of interest in the tomogram, one thing that you can do is one thing that is called subtomogram averaging. For example, thinking in ribosomes, the cell is completely full of ribosomes, okay? And we know the structure of this ribosome, but we don't know how is the structure of this ribosome inside the cell. So the thing that we can do is we can make the tumor run of the whole cell. We are going to make the segmentation of different ribosomes inside the cell, and we can put all this information, all this ribosome together to try to increase the signal and also to try to fight with this missing wedge. Because in this protein, or in any cell, the ribosome was located in different orientation, okay? So you have different three-dimensional reconstruction of this ribosome and with different missing wedge. If you put all these ribosomes together, the missing wedge of this one is full with the information of another ribosome. So in this case, you are going to obtain two things. You are going to be able to manage this missing wedge and obtain the information of the whole sphere, and you are able to increase the signal. You are able to see also the high resolution of this protein, ribosomes, nuclear complex, whatever, of this protein inside the cell. And this information is very powerful because you are not only knowing the structure and the function of this protein, you also knowing how it's going to carry out this function in different part of the cells. Maybe the ribosome closer to the nucleus is in a different conformation than far away in the reticulum endoplasmatic. So they give you information about the structure, but also they give you information about the context of this protein inside the cell. Okay. One video, if it works. This is the field series. Basically, the idea is they are going to inclinate the sample to obtain different images, put all this information together, and after that, another tilt series from a different region of the cell. And after that, you are able to obtain a few-dimensional reconstruction of the region of interest of your cell. In this case, these guys are interested in the vesicle trafficking in chloroplasts or something like that. So they are going to focus in this region. They are going to extract this information and continue the study, okay? That's the idea. Okay. Fine. As I say at the beginning of the talk, cryo-EM in the past was covering the gap between the light microscopy and the high resolution techniques, okay? But in the last 10 years, going to occur a lot of improvements in the microscope, but also in the software that we use to make the processing of the data that was able to push the resolution in cryo, single particle, and also in cryo-electron tomography, and was able to arrive to high resolution, okay? So just to introduce, here's the data, and the number of PDBs, that is the structure models of protein was increased a lot using cryo-EM compared with the results in the past. And the different changes in microscopes this was the most important, was the development of direct electron detectors. Basically, in the past, we are using CCDs, like the digital cameras, that are going to, the electrons after arrive from your sample is going to arrive to your camera, and in the CCD is going to change to photons, and electrons after photons, and this photon is going to move again to detectors to obtain an image. The problem of this detector is that they are very slow, and the pictures that you are obtaining is something like that. There is like blurring there because everything is very slow and start to move, and you are losing the detail. You are losing the high resolution information in these detectors. So 10 years ago is going to appear the first direct electron detector, basically the electron that arrive and is detected for the detector. There are not any change. So the good thing is that they're very, very, very fast, and they are going to be able to retain the information. You are going to see all the proteins in detail. So with these changes, we are able to arrive to high resolution in cryo-EM. The second thing is that when you have a protein, you have a protein in this picture that are in very different orientation, okay? But also remember that the protein is not rigid. So there are a lot of different conformation with a lot of different orientation. So the amount of information that there are in one image is a lot. And if you are working with 10,000, 20,000 images, 20 million of particles, the software was not able to solve this gap, okay? So very smart people start to work in this problem, and they're going to design different algorithms to try to manage this big amount of data. And they was able to isolate the different conformation of the protein and the different orientation to try to solve the structure of this protein at high resolution. So all these things are going to, nature methods say that the cryo-EM with 80 years of history was the method of the year of this year. In 2017, Jacques Boucher, Joachim Frana, and Richard Henderson are going to receive the Nobel Prize, and it's going to occur one thing that is called the resolution revolution, that all the structural departments, all the universities start to buy cryo-electron microscopes with this detector, a lot of them, and also here. In Barcelona, 5 big institutes from here, the Barcelona Molecular Biology Institute, the Alba Synchrotron, Institute for Research in Biomedicine, the Center for Genomic Regulation, and the Tony March University. They're going to put money all together. They're going to ask for more money to the European Union to buy a high-end microscope, okay? So this microscope was installed in the Alba Synchrotron, that is very close to here, in the Autonomous University. And right, I think it's here, is going to make a new building. That was the electron microscopy center at Alba. That was capacity for 3 microscopes. This is the life science one, and these 2 are for material science, okay? This is the microscope, the control room, and there is a small lab. This is how it looks like the room before to install the microscope. One thing that is very important also to describe is that it's so important, the microscope and also the room. You need to completely isolate it for everything. Don't change in temperature, acoustic isolation. You cannot move anything. And even the magnetic fields, we have to install some sealed with magnetic fields to protect the microscope and to obtain the better data. Okay, and after that, to isolate the microscope, the manager of the microscope is in another room that is closer to the microscope, when you have all the things that we can use to make the single particle analysis and tomography data acquisition. And also, we have all the things that you need to make the first step of the processing of the data, to see how it looks like your data, the quality, the proteins, blah, blah, blah. This is the sample, the small lab that we have to prepare it. And just to give you an idea on how far we can see the proteins, this is a reconstruction that we vitrified here in Barcelona of a protein that is called apoferritin, that is like the benchmark that all the microscope managers use to see how good your microscope is. And here a 2-point Armstrong resolution when we are able to see the side chains of the protein. Okay, so no bad. We opened 1 year ago, and we have a lot of users from different places, different hospitals, blah, blah, blah. We have a lot of data, and we have the first publication, okay? We opened 1 year ago, and we are in a collaboration with Professor Thomas Uri from the CRG and Professor Oscar Jorger from the SNIO, and we are going to solve the structure of this complex between the gamma and microtubule that I explained, and with this result was the first publication of the platform that was published in Science this year. And just to give you an idea about the publication, the microtubules is the protein filaments that are in cells and basically are forming for protofilaments that are polymerized the heterodimer alpha beta, okay? So there are different number of protofilaments in microtubules in vitro, but in cells, all the microtubules are forming for 13 protofilaments, okay? So the protein that was using as the template to start the nucleation of this microtubule was called gamma-tube. That is this protein that has a shape like a cone, okay? 2 megadalton protein that is forming for different GCP's protein and the gamma tubulin that is going to act as the template to start the nucleation of the microtubule. Okay. In this case, the structure of the gamma tube was solved 2 years ago, and showing that there are 14 tubulins and the microtubules are 13. The question is that this gamma tube is looking like an open conformation, likely an inactive conformation that was not able to polymerize. How this protein is going to be able to move from here to 13 tubulins to start the microtubule nucleation is one question that we try to manage using cryo-EM. The first thing, this is the question, how we can start to make the polymerization, the nucleation of the microtubule. So the first thing is that we need to prepare microtubules with gamma tube, that are very, very short and in a big number, because we need a high amount of that one to make cryo-EM. So the first thing that was developing for Claudia Brito, the CRG, was trying to see to obtain the big numbers of microtubules with gamma tube and shorter as we can. So the thing is that here is a confocal microscope that we are going to show you the microtubules. And in the case that in this mutation of the tubulin, they are able to increase a lot the number of microtubules moving from this one to this one, okay? Also, the concentration of this mutant of tubulin are going to increase the number of this complex, the gamma tubules, a lot. In this case, when we compare the number of tubules or the nucleation rate between the mutant and the wild type, is going to increase like 2 orders, okay, at the same concentration. So, okay, we are able to obtain a lot, and now we also need to prepare shorter as we can. And there is one protein that is called DARPin. It's located at the plus M of the microtubule. So basically the idea is, okay, we are going to add in gamma tube, we are going to start the nucleation with this mutant, and we need to add the DARPin protein that basically the idea is going to curve the protofilaments, avoiding the elongation of that one, okay? So using different concentration at different times, we are able to decrease the length of the microtubules until we obtain a very, very short complexes, okay? So after that, we move to the sample preparation. That was very hard in this case because at the beginning, there are no microtubules at the host. All of them are going outside and they are very, very long. And using this approximation from Claudia, we are able to obtain a lot of microtubules inside the hole and obtain here in Barcelona a first reconstruction, a low reconstruction of this complex, prepare more grids, and move to the next step. All the processing of this data was carried out from Marina Serna, from the Oscar Yorca lab in Madrid. We prepared a lot, I don't remember, maybe 20, 30, 40 grids, and we sent all these grids to the higher microscope that was installed in Bilbao, Institut Biofísica. They acquired 44,000 of movies. They Marina processes in a very, very long job all these different data, and that's the results that obtained. Marina was able to isolate 5 different states of this complex, okay? All different states are different in the size of the gamma tube and also in the length of the microtubule, okay? In 2 of them, in 2 close conformations, we were able to arrive to high resolution and obtain this reconstruction when we are able to solve the question that we are talking. That's the gray one. Okay. The gray one is the open conformation where there are 14. Maybe it's easier in the next one. Yeah. In the open conformation, if you remember, we have 14 and we need to move to 13 tubulins to start the polymerization of the microtubule. Okay. In this that we call early close conformation, the thing is that the 14 tubulin is going to put it over the number 1. Okay. This GCP is put it over the gamma tubulin and is going to reduce the number from 14 to 13. Okay, the 1 and the 14 is one above the other. The thing that is going to change is they're going to change they are going to change in the conformation of the GCP. Ahí no se ve. From this position to this position, And all these things together are going to move to an open conformation to a closer conformation, okay? And all these things is only changed for this small electron density. Marina obtained in the three-dimensional reconstruction was observed that there is extra density just here that nobody knows what is that. But the thing is that this protein is going to act like a latch. Basically, it's going to interact with the alpha tubulin of the protein 1 and the 2, and are going to make it smaller, okay? And this is the second conformation that we obtained. That basically the idea that we are able to see here is that when with the strata that we solve, this is a reconstruction of the microtubule. When we put over, we fix exactly with 13 monomers. And in the case of the upper conformation, it's like an inactive. So there are not a way to start the nucleation of the microtubules, okay? So yeah, that's all. Thank you for your attention. Any question you can ask.
So do we have some questions from the audience? So I will start. So we are immunologists and we're like to see, for example, let's say the interaction between two proteins. One could be an antibody.
There is one thing that I want to show you today, but when he arrived, told me that the talk today is in streaming, and I have some unpublished results for between protein and FIBs that I cannot show you.
No, but my question is so are the limitations related to the size of the protein? For example, an antibody recognizing another protein, that will be a problem.
The
it's too small, or how is that?
Smaller is the protein, challenging is the project. Everything that is below 150 kilodaltons is challenging, but it's not impossible. I mean, the slide that I would like to show you, but I finally had to put it, it's a complex with a membrane protein of 45 kilodaltons that is called CD36, that is rated with a lot of different things. And I have the complex of this protein with FIB that are 45. So in total it's 90.
So, but that will be the kind of project that we may be interested. So that will be feasible in a way. And then I'm wondering, so imagine that I want to do something like what you just mentioned, and how would be that how can you manage that? So I come to you, is it a service? Is it just purely scientific collaboration? How does it work?
There are the platform of the cryo-EM platform that was installed in Alba. It's a service that you have to pay for all, but the expertise is there, so you can bring with your protein in solution, and we are going to prepare the samples. We are going to make the screening. We are making the data acquisition at the first steps of the processing. After that, when you want to looking for a three-dimensional reconstruction at high resolution, we can help you, we can teach you, but we cannot do it for you because we are only two at the platform. But there are a lot of people, I mean, in Barcelona, there are not cryo-EM community 1 year ago. And right now, everything starts to move, looking for collaboration like these guys, or start to learn and teach. So.
But I guess a project like the one that you showed, it's really complex because
this one is maybe one of the best, of the more complex, obviously. But
but it's something like more simple. Then I would have also, I'm curious to know how much would that take. So
I have a lot of users that are looking only for the epitope between the a lot of them from the IrsiCaixa, from the clinic. A lot of people that are going because they are only interested in knowing which one is the region of interest between the FAB and the project, and they are starting to, I mean, they are working.
Yeah, but is it fast or does it take some time to?
3 months.
3 months, okay. So
yeah, that's maybe sometimes faster, sometimes slower, but I think more or less 3 months from you arrive to the door of the platform until you have a three-dimensional reconstruction and medium resolution.
So do we have questions from the audience? Yeah, over there.
Do you think this technique can be improved or to get more resolution, or is
It's improving. Right now, most of the things are improving in software, and now our people are using machine learning or this kind of stuff are improving the picking, improving the signal, and improving the algorithms. But in terms of sample preparation, that is the main limitation right now. There are not any robot or anything that can make better than the guillotine. It's technology from the 80s, but it's the best that we have.
Okay. No more questions? Okay, so thank you very much.
You're welcome.
So we're going to end. Eva was supposed to finish with the session. I think it was quite interesting, and look forward to see you at the next meeting. Okay, thank you. Thank you so much for coming.
Okay, to right now the
Tendrá que doy otro. No para de más larga.
Porque realmente proteínas con las que estamos trabajando, que definen algunos de los aspectos técnicos. Hay una que tienes, creo que aparte de la pero luego eso depende de los dos últimos. Este 229. Y al lado, no solamente estamos trabajando.
Bueno, porque los estudiantes
miedo.
Les había dicho que a Jiménez
Sí, sí, también. Y hemos hecho justamente anticuerpos para ella. Pero justamente voy a mirar por unas horas.
En todas las pero sí que normalmente se hace
Pues me lo miré, precisamente hace años, que trabajamos nosotros, que es el Y,
prepara una cosa así.
yo había visto entonces que estaba en las 6, el perfil. O sea, que estaba ahí. Pero es interesante porque está muy por ahí se produce.
Pero alguien ha puesto porque
creo que sí que hay posibles. O sea, es que o sea, yo no pero sí que el paper es todo. Yo creo que sí que hay de técnica y si no se puede meter los datos.
No estoy seguro, pero sí que había. También es el sistema de